We'll get started in just a couple minutes. Good morning, everyone. We'll go ahead and get started now. My name is Rochelle Seitz. For those of you who don't know me, I'm the director of the VIMS REU Summer Intern Program, and just wanted to welcome everyone and say thanks so much for joining us, whether in person or via Zoom. And we are so happy today to have many of our REU interns in person to give their final presentations. And so it's been I think a long road this summer, getting all of the data collection and kind of rolling with all of the changes that came about with our changes with COVID restrictions, and finally the ability to have everyone come in person. So we're really happy that we're able to do that. I can't thank enough the folks that have been involved with this REU program all summer. So all of the mentors, thank you so much for all of the work that you've put in with these students. I know it's been a little bit difficult and trying at times doing things over Zoom. Trying to teach statistical methods over Zoom is not the easiest. and also certainly Grace Massey, who is our program coordinator, who's dealt with everything from getting the students their paychecks to getting travel arrangements when necessary, and everything in between. And then also our TA, Stephanie Peart, who has done a great job with all of the organization for all of our Zoom meetings and getting the students prepared for today's presentations as well, with comments on all their practice presentations and everything. So if we can have a round of applause for our mentors and coordinators. Yay. Thank you so much. So what we are going to do is we will have student moderators for our 2 sessions today. And each of our students will be given a 20-minute time slot. And the talk should be about 15 minutes. And then we'll have 5 minutes for questions. And we'll pass around the microphone if we have a question from the audience. And we can also monitor the chat feature in Zoom, right, Steph? For those watching via Zoom. And so our moderators will give a little hand signal to the presenters to let them know when they've got just 2 minutes left, and then again when they're at their 15 minutes. So I think with that, we can go ahead and get started with our moderator for our first session, who is going to be Nihal Guennouni.
Thank you. Hi, my name is Nihal Guennouni, and I am one of the REU interns here at VIMS. I just wanted to get started and introduce our very first presenter. His name is Duncan Campbell. He attends William and Mary, and his mentors are going to be Dr. Song and Stephanie Wilson.
Thank you for the lovely introduction, Nihal. All right. So I'll be presenting today on nitrogen cycling in seagrass beds and changes in seagrass bed community structure during restoration. So I'm going to briefly cover seagrasses and the ecosystem services they provide. I'm going to look at sediment microbes and their function in seagrass beds, and then how our lab analyzed the community structure and denitrification of these seagrass beds, and then what we found. Seagrass beds, for some broad overview, are a extremely valuable habitat. Seagrasses provide a large number of ecosystem services to the marine ecosystems around them. They provide nursery habitats for larval fish and invertebrates, especially blue crabs in the Chesapeake. And seagrasses are biogeochemical hotspots. They are excellent high-density carbon sinks, and they cycle nutrients at a much higher rate than bare sediment can. Human activity has decreased coverage of seagrass beds throughout the Chesapeake Bay, but restoration efforts have been successful. In the last 40 years, we've drastically increased coverage of seagrass beds throughout the bay. However, we are still below our target coverage, and restoration efforts require more analysis so that we can increase our efficiency. Of the two species that we focus on for restoration efforts, we typically consider Zostera to be more beneficial to the bay. Zostera marina, known as eelgrass, is the dominant species of seagrass in the Chesapeake Bay. It prefers colder temperatures and grows more densely, taller, and puts deeper roots into sediment beds. And so it can support more organisms compared to our other dominant species in the bay, Rupia maritima. Rupia prefers warmer temperatures. It's typically found in more southern environments than the Chesapeake. It grows less densely, and the plants are smaller. In addition, Zostera tends to be more annular. It returns more regularly and in healthy seagrass beds compared to Rupia beds. And so in the bay, when we are dealing with issues like harmful algal blooms, one of the things that can explain a harmful algal bloom is nitrogen runoff. Nitrogen runoff is an issue in a number of different environments, and the bay normally compensates, marine environments compensate for nitrogen runoff by undergoing a denitrifying process, either denitrification, in which sediment microbes consume nitrate, the common molecule in fertilizers, and produce nitrogen gas, or the dissimilatory nitrate reduction to ammonium pathway, DNRA, which converts nitrate into ammonia, which can be recycled in the sediment. Typically, we find denitrification occurs with DNRA in seagrass beds. Seagrass beds, because they have higher biomasses, support larger communities of sediment microbes. They are known to export oxygen out of their roots. Plants produce oxygen through photosynthesis. Seagrass puts that oxygen in sediments. The sediment microbes react to this change in oxygen concentration. And so in seagrass beds, we'll typically undergo higher rates of denitrification because the oxygen makes it more favorable. We typically see slightly higher rates of denitrification than DNRA in Zostera. And in Rupia, we tend to expect more denitrification relative to, more DNRA relative to denitrification compared to Zostera. Both will have higher rates of nitrogen cycling than bare sediment. We set out to characterize the sediment microbial community that is undergoing these processes and to measure these nitrogen cycling rates. Again, we expected to see different community structures between the two species, and we expected to see that Zostera beds would have higher rates of denitrification. we had a number of treatments through the SAV program, the VIMS program that is helping restore seagrass beds throughout the Chesapeake. And so in the Lynnhaven River, where we were looking, we took bare sediment samples as control. We also took sediment samples from seagrass beds planted in the fall. There were Zostera beds and Rupia beds planted in the fall. We refer to these as our Zostera and fall Rupia samples. And we took sediment cores from plots planted in the spring. Our spring Rupia treatment, as well as a planting of Rupia where they were shocked with fresh water before germination. This simulates the conditions found in the wild, slightly more than just planting Rupia germinated in saltwater wood. And we took our samples in October of 2020, in the fall, in April of 2021, our spring sampling, and June and July were our summer samplings to coincide with the peak growth rate for the two species, as Zostera beds reach their peak growth rate before Rupia beds do. We selected our sediment cores from the top 5 centimeters of the plot, and we created a slurry from some of the sediment, which we then incubated with 15N nitrate, a stable isotope of nitrate that we can use to accurately measure nitrogen cycling in a set time period. This allows us to differentiate from nitrate that would already have been in the soil, the sediment. And we underwent 16S ribosomal RNA sequencing for use in bioinformatics pathways. We used the DATA2 pathway, along with the phyloseq pathway and the vegan pathway for community analysis. And we were able to also perform a metagenomic analysis using the PyCrust2 pipeline. We found we were able to draw a number of comparisons between our microbial communities using the 16S ribosomal RNA sequencing, and we were able to measure our denitrification rates and draw a number of comparisons there. This here is a PCOA. It is a plot that shows us similarity of samples. And in this case, this is our microbial community. And so the points represent different samplings, a single sample in which we have done sequencing, and we are able to identify a single community. You will notice that there are clusters based on color. The colors represent our seasons. So everything red is a sample that we took in the fall. As you can see, there are distinct patterns in our seasons. Our seasons have clustered. And what this tells us is that there is a significant seasonal effect. Our seasons were having a strong explanatory effect on the community structure. So looking at our individual seasons, we find there is no significant difference in our fall treatment. Our fall samples are all very similar. And this makes sense because the plots had not started growing in the fall when we took our sediment cores. So everything should be very similar to our control. You'll also notice that there are only 3 treatments on this plot. This is because we had not yet planted our spring treatments, and so we could not collect data for them. In the spring, we have all of our treatments planted, and we see some clustering, but not extensive clustering. The green triangle there is our 3 Zostera samples. They cluster to an extent away from our Rupia samples. For example, the purple bubble up there is our spring Rupia freshwater shock samples. They were highly clustered, but overall you will notice that there is a distinction to an extent between these samples. And so when we performed statistical analysis on these samples, we found there were significant differences between Rupia samples and our Zostera samples. This means that our Zostera is growing and it has managed to change from our control and from the rupia beds that have developed so far. In the summer, our sediment plots have finally turned into seagrass plots. We have coverage of seagrass for all of our samples. And excitingly, we have clustering. Our controls are in their nice little bubble down at the bottom on this plot. But you notice our Zostera has moved further away from our rupia samples, and our rupia samples have clustered tightly together to the side of this plot. This is very exciting for us. And we were able to look at denitrification, and we found significant effects based on season. This plot shows our denitrification rates between seasons, our fall seasons, our spring, and our summer. The side is the average denitrification rate occurring in a 1 hour incubation. So over the course of 1 hour in our lab, how much nitrogen was processed. And we found that we did see differences from our fall plots. Our fall sampling was different from our spring and our summer. And our Zostera in the summer had the most denitrification. This was a significant effect. And our fall rupia had the most denitrification occurring in the spring. And so we concluded that we have seen unique microbial communities develop. We have seen significant seasonal variation in our denitrification, and our seasonal variation has been observed for our microbial communities. And so what we conclude from this is that our seagrass species affect the geochemical condition of the sediment. Planting seagrasses changes sediment. We found that denitrification rates were higher in Zostera beds. We expect this to be due to higher root exudate, higher oxygen export from the roots into the sediment production. And we found that there was higher denitrification rates in our rupia beds in the spring in some treatments. Our fall rupia has been growing the longest of our rupia samples. And so we expect that our fall rupia, which has had the most chance to grow and can grow faster in the spring than Zostera, may have had more effect on the sediment than our Zostera at this point in our sampling, and so had caused the highest denitrification in that sampling. And so we plan to continue monitoring our seagrass beds. We can plan to continue watching these seagrass beds as they are restored, and we hope to see more changes. Of course, restoration takes time. You can't grow an entire seagrass bed across the entire bay in 1 year. And so we also plan to compare to other areas where we are restoring things. These samples were taken in the Lynnhaven, as I mentioned, but we are also monitoring plots in Brown Bay and other locations throughout the bay. And we expect to see differences based on location as well. I'd like to acknowledge Dr. Song's lab, Dr. Song, Stephanie Wilson, and his lab coordinator, Michelle Cochran, who was incredibly helpful anytime I was dealing with any experiments, and Dr. Sam Fortin, who was working as a postdoc in the lab when I started and helped me a ton with all kinds of our analysis. Hunter Walker ran a number of nitrogen composition analyses for us. I'd like to thank the REU program, Dr. Seitz, Dr. Massey, our TA, Stephanie, and the SAV program, Dr. Chris Patrick and Dr. Annie Hessel, who collected the samples for us and were super helpful. They provided pictures. They helped us think about the data. It was just, we couldn't have done it without them. And of course, I'd like to thank you, the audience.
All right, thank you, Duncan. So now it's time for any questions. And if you are over Zoom and you guys have any questions over there, feel free to put it in the chat or just chat that you want to turn your microphone on and we can have you guys ask those questions there. All right, we have a question from the audience.
Nice job, Duncan. My question is about your denitrification results. And I'm trying to remember back to your hypotheses, but from your research questions and hypotheses, were you expecting differences in denitrification among the different seagrass treatments?
To an extent, we expected a difference based on species. We had hoped to see some difference based on treatments, but we think that the seagrass beds were not developed enough for that to be appearing yet.
Mm-hmm.
Yeah,
so it seems like the biggest differences are seasonal,
yes,
versus among the seagrass species and your control. But in summer, you did see what looked like distinct microbial communities among the treatments. So how do you put those two ideas together? What do you think is going on?
It's difficult to say. We had a relatively low quantity of replicates for these denitrification results. And so it is difficult to draw absolute conclusions at this time. We do see some differences, but they're not always statistically significant, and so we didn't want to draw any conclusions falsely. Does that answer your question? Okay. Okay, sure.
Yeah. If the results you found would still exist if you increased your replication, what do you think is going on if the microbial communities differ but denitrification rates don't differ among your seagrass types?
We think most of it is just due to how much seagrass coverage has occurred so far, how much the seagrass beds have developed. We just think that the seagrass beds aren't mature enough to be really changing the composition of the patterns of nitrogen cycling in the sediment. But it's also, nitrogen cycling is a complex, multifactorial process, and it's possible that the temperature is having an influence, that these plots were in the same location, so we don't think that it should be an issue, but there's always some possibility there. Or maybe the Rupia samples simply just don't differentiate enough. It's possible that this is just simply the natural occurrence and that we have to do more research to really determine why.
Great, thank you.
All right, we have a question from Amber Hardison. Sorry if I didn't say your last name right. She said, "Great job, Duncan. I enjoyed your clear presentation. Did I see correctly that the highest denitrification rates were in the fall when the seagrass beds were not yet established? Any idea why that may be?"
Hello, Dr. Hardison. It's nice to hear that you're on. This is true. We did see high denitrification rates in the fall. We thought that might just be a function of the season. We didn't want to draw any conclusions about why that would be until we had a chance to collect more samples and see what our controls do year to year. We didn't have a perfect explanation for this yet, and we are still considering it. We are still working on some analyses of dissimilatory nitrate reduction, and we think that we may be seeing some changes there. But since we haven't managed to collect all of that data yet, we could not include it in this presentation at this time.
All right, thank you, Duncan. We have time for one more question, and so it seems like we have one from the audience, from Amanda, so go ahead.
Hi, Duncan. Nice job. My question is for the PCOA plots. I might have missed it, but can you just tell me what a PCOA plot is? And we were looking at just like clusters that we're grouping together, right?
Yes. So a PCOA shows similarity between samples. And if you want the math that it shows the difference in the eigenvectors of the samples calculated based on a number of different analyses. To not turn this into a math lecture, we use a number of calc tricks and stat tricks to decide what a sample looks like in a 3D space, and then we measure how far away it is, and then we plot that in 2 dimensions. And it's a headache, but the general answer is that if you look at the PCA or the PCOA and you find how far away 2 samples are, that's how different they are. Yeah.
All right, good job, Duncan. If everybody wants to give him a round of applause. Let me grab a seat. All right, and now we're going to introduce our next student presenter. And we are pulling up her presentation right now. Her name is Maya Casey. She attends Haverford College, and her mentors are, her mentors are Dr. Juliette Smith and Vanessa Strohm.
Thank you, Nihal, for that introduction. Hi, everyone. I'm Maya. I go to Haverford College, and this summer I looked at the impact of prey and temperature on the emergence of the toxic dinoflagellate, Dinophysis. So like I said, our organism of interest was Dinophysis. Dinophysis is a genus of algae, and it is a dinoflagellate. And Dinophysis has been known to produce a number of harmful algal blooms, also known as HABs. There are a lot of subspecies within Dinophysis. We were interested in the genus more broadly. Particularly, we're interested in Dinophysis because of its ability to produce toxins. 2 of those toxins are okadaic acid and Dinophysis toxin. When these toxin levels reach a certain threshold, they can cause a condition called diuretic shellfish poisoning. So essentially, when toxin levels in shellfish get beyond a certain threshold, anyone who consumes these shellfish can then become sick. So Dinophysis is found all over. It's actually found internationally, but it can also be found in all the US coasts. And as you can see, it's found on the West Coast, East Coast, and Southern coasts. And the way that this is tracked is actually through labs and PIs, but it's also been tracked through news. So news of DSP cases, as well as shellfish bed closures. I would like to point out that most of the locations shown on this map have had cases of DSP or shellfish bed closures, but we actually have yet to see that in the Chesapeake Bay. And that's actually a big question for us. What are the factors that influence high levels of Dinophysis in the Chesapeake Bay, and why do we have yet to see that? So one of the factors that we're interested in is prey. Dinophysis is a mixotroph, which essentially means that it's able to survive on its own for extended periods of time just with light. However, it'll eventually die if it doesn't have that prey available. One of the key prey groups for Dinophysis is Mesodynium rubrum, which is a ciliate. If I can direct your attention to the schematic on the bottom here, on the far left, you see a cryptophyte. Mesodynium, which is a ciliate, consumes that cryptophyte, and then Dinophysis latches onto Mesodynium and sucks out all the cell contents. One of the key cell contents that's passed through this food chain is the plastid, which is also just a chloroplast. The plastid is really important for a number of reasons. One, Dinophysis can't photosynthesize without it. Dinophysis also needs the plastid in order to produce toxins and grow. So in other words, Dinophysis can't become a HAB without the plastid. And then eventually shellfish will feed on algae and toxic algae like Dinophysis, and that's what contributes to diuretic shellfish poisoning. I would like to note here that this chain has mostly only been studied in a culturing context and has yet to be universally accepted in nature. In terms of temperature, I was interested in looking at temperature for a number of reasons. Dinophysis is a type of algae, and algae is likely to be sensitive to changes in the environment. And the Chesapeake Bay is a pretty dynamic system, so I thought the temperature could probably have a pretty drastic effect on when Dinophysis blooms. Dinophysis and Mesodynium, or the temperature has been studied in regards to Dinophysis and Mesodynium, and what they found was at low temperatures, so the lowest temperature here, which is marked in yellow, it's a little bit hard to see, but on the graph I've outlined it in black, so it's a little bit easier. They found that Dinophysis had a really low cell concentration and actually had a really slow growth rate. They found that Mesodynium had a really high growth rate. At a high temperature, which here was 32.5 degrees Celsius, marked in red, they found that Dinophysis had a really high cell concentration and Mesodynium actually died really quickly. So to summarize what the temperature trends were in this paper, generally as temperature increased, Dinophysis cell density increased and Mesodynium cell density decreased. This brings me to my objectives. So I have two objectives. My two objectives were to investigate prey availability and patterns of Mesodynium and other ciliates in the York River. I'd like to point your attention to other ciliates. So we do know that Mesodynium is a key prey group for Dinophysis, but we're curious to see if there's other ciliates that behave similarly. We also want to consider how temperature could be affecting Dinophysis, Mesodynium, and ciliate abundance. So my hypothesis in regards to prey availability was that high concentrations of Mesodynium and other ciliate prey groups would precede large Dinophysis species blooms. This is solely based on the assumption that you need a lot of prey in order to attract the predator. In terms of temperature, I predicted that Mesodynium and other ciliate blooms would occur in colder seasons prior to large Dinophysis blooms in warmer seasons. And this was based on the literature. So now getting into my data collection. I used primarily the imaging flow cytobot, or the IFCB, which is an instrument that uses a mix of high resolution imaging and flow cytometry to take images of, it takes up water samples and then captures images of phytoplankton cells. Our IFCB was deployed off the VIMS pier in the York River for several months at a time throughout the year 2020. And so on the left, you can see our IFCB on a lab bench. And so that's what it looks like when it's out of its cage. There's a lot of systems in place to prevent organisms growing within the IFCB and to prevent potential contaminants. Potential organisms that can grow on the IFCB are shown on the left there, or right, I guess if you're facing. But essentially that's our IFCB after 1 month of deployment. And there's tunicates, barnacles, all sorts of biofouling that grow on the outside of that cage. I also used NOAA's tides and currents temperature data. My second methodology was the part in MATLAB where I'm looking at the images that are produced from the IFCB. The IFCB takes up to 10,000 images every hour, so I had an ample data set to work with. So there are 126 species in our classifier currently, and we used a random forest machine learning algorithm. So essentially what I would do is I would open my screen and this is what I would see. On the left, you see, the left and the right, you see columns that include different algal species and other species found in the Chesapeake Bay. And then what I do is I go through and I classify each image into whatever category it falls into. And our goal with this is then using the machine learning algorithm, we hope to be able to train the IFCB so that it can do it on its own. So my data came from what the IFCB was able to classify on its own. So getting into my results now. This is an abundance plot and it shows Dinophysis and Mesodynium throughout the year 2020. So how you read this is on the left blue axis, you see Dinophysis, which you can see that the cell concentration is really small. So the cells per mil was measured by however much water was taken up in the IFCB. They counted the number of cells and then they divided it to make it per 1 milliliter. You can see that Dinophysis has a really, really small cell concentration. On the right axis, which is marked in red, that's Mesodynium, and it's measured the same way. But you'll notice that they have very different scales, and that's just due to the amount of cell concentration for each one. We don't really care as much about the scaling because we're only looking for peaks and when these species were at their highest. So in other words, we care about when they peaked, but not how much they peaked. So what we found was that Mesodynium consistently peaked after Dinophysis. We also looked at Mesodynium versus ciliates. So just to pause here, like I said in my objectives, we were comparing Mesodynium to other ciliates to see if they behave the same way. So I was interested in looking at our ciliates category in our random forest machine learning algorithm, which encompasses 18 different species of ciliates. and that's excluding Mesodynium. So Mesodynium is a ciliate, but it's not included in this broad ciliates category in order to create this comparison. Ciliates are shown in the bottom graph and they're marked in green, and the top graph is the graph that you saw on the previous slide. So what we're looking for here is to see if that top red line matches that bottom green line. And what we found was they both had peaks in April, different amounts of how much they peaked. But then ciliates peaked again throughout those warmer summer months. But interestingly, they did have very similar patterns in the colder end of the year months. So going back to my original hypothesis, I had said that Mesodynium and other ciliate prey groups would precede large Dinophysis species blooms. And we actually found this to be wrong. We found peaks of Mesodynium were found after large Dinophysis species blooms. Another important finding was that Mesodynium had different abundance patterns than other ciliates. So now getting into my second variable, which was looking at temperature. That top graph was manually created based on the data from NOAA's database. It shows a average daily temperature throughout the year 2020. That bottom graph is the graph that you've seen before, which is the abundance plot of Dinophysis versus Mesodynium. And what we were looking for here was to see in what months and at what temperatures Dinophysis was at its highest abundance. And what we found was that Dinophysis peaked in March and then again in the summer and early fall. We also looked at this for Mesodynium, and we found that Mesodynium peaked in April and then peaked again at the end of the year. We looked at this for ciliates as well, and we found that ciliates actually just peaked throughout the year. There wasn't a clear trend or pattern. To better visualize the data, I created a correlation plot. The way this is read is you go across the row and down the column. In that box where they meet, there's a circle. The size and the colors of the circle represents the correlation between those two variables. So in terms of temperature and Dinophysis, we actually found that there was no correlation because you can see there's no circle there. In terms of Mesodynium and temperature, we found that there was a slightly negative correlation between those two. We also found a slightly positive correlation between ciliates and temperature. So again, going back to my original hypothesis, I had said that Dinophysis cell concentration would increase with temperature. And we actually found that this was incorrect. Dinophysis and temperature didn't have any correlation. I had said that Mesodynium and ciliate cell concentration would decrease with temperature, and this was partially correct. It was incorrect in that ciliates and temperature actually had a slightly positive correlation, but Mesodynium and temperature had a negative correlation, meaning as temperature increased, Mesodynium cell concentration decreased. I just want to pause here and emphasize, this is only 1 year of data. So this was just for 2020. So these trends that we were seeing were just for this year. And so we can't really say conclusively that this would happen in every year, especially given how dynamic the Chesapeake Bay is. It was very interesting though to see these results, especially that temperature and Dinophysis had no correlation. But in terms of the ciliates encompasses 18 different species. So we can't, if we were to break up that group into smaller subspecies, we might see different trends. So getting into my conclusions, Mesodynium does not clearly predict Dinophysis species blooms. Because Mesodynium was consistently found after Dinophysis, we can't really use it to predict when Dinophysis is going to occur because it almost always occurred after Dinophysis. Another important finding was that Mesodynium and other ciliates had different abundance patterns. This is important for a number of reasons. If you'll remember in my initial hypotheses, I tended to group together Mesodynium and ciliates to have the same trends. So I would say Mesodynium and ciliates are going to do this. And I didn't really think to separate them, but what our results showed is that I actually definitely do need to separate them when I'm making hypotheses. And also, like I said, even separating ciliates into smaller subcategories would be valuable. We found that temperature did not appear to be an influential factor in the growth of Dinophysis. That's not to say that temperature isn't impactful at all. Dinophysis is a type of algae. It is going to prefer certain temperatures over others. Temperature just didn't appear to be a driving factor. And this could have happened for a number of reasons. We know that in the environment, there's a lot of factors working together. So it could have been temperature and salinity together could affect when Dinophysis occurs, but temperature on its own was not the most impactful factor. And finally, temperature may be influential in the emergence of Mesodynium blooms. We did see that negative trend, so we could potentially expect Mesodynium blooms in those colder seasons. In terms of where to go from here, so like I said, in the environment, you have a lot of factors working together. We found that temperature wasn't a key driving factor, which means there's likely other factors working to influence when Dinophysis blooms. So some of these could be salinity, turbulence, water quality. And the way that we can test this is in culturing experiments. The picture on the bottom right is me actually helping Vanessa in our lab with her turbulence experiment. So she's currently looking at the effect of turbulence on Dinophysis and Mesodynium. Another future direction could be looking more at why Mesodynium and other ciliates have such different abundance patterns. Like I said, the ciliates category was pretty big, so breaking that apart would be valuable. I'm also just curious to know if there's some biological difference between Mesodynium and these other ciliates that makes Dinophysis more attracted to Mesodynium than the other ciliates. Finally, we hope to be able to strengthen our IFCB classifier through a new classification network. So our IFCB is a really valuable instrument because it's able to detect cell concentrations that are really small. So we can actually use it to create an early warning system, meaning that it can detect really small cell concentrations of a harmful algae, and then we can predict when a HAB might emerge. So we're actually switching over to a new classification network now, which will be able to differentiate on a more individual level between species. So we won't have that block of ciliates, we'll have each individual species. Finally, I'd just like to stress the importance of my work. So Dinophysis, this is an article from a little over 2 weeks ago about a shellfish bed closure that happened in Washington State from Dinophysis toxins emerging. And so shellfish bed closures in cases of DSP are becoming increasingly more relevant. And so we need to be able to track these HABs and identify when they're going to occur in order to take the appropriate precautionary steps. I'd like to give a special thank you to the VIMS HAB Lab, my two mentors, Vanessa Strohm and Dr. Juliette Smith. The REU program coordinators, Dr. Rochelle Seitz and Dr. Grace Massey, and our REU program TA, Stephanie. I'd also like to thank all the funding that I received to help me with this project. I'm now happy to take any questions. Also, here's some pictures from our IFCB recovery, which was one of the highlights of my REU experience because I was able to see the IFCB in action and get some field experience. So thank you so much for listening.
All right, thank you, Maya. So as you've heard, we are opening it up to any questions that the audience may have. And again, reminder for Zoom people, just say question in the chat or type your question out. All right, we seem to have a question in the audience from Duncan.
Great presentation. Really enjoyed it. You can hear me, right? Yeah. So my question was you had the Mesodynium responding to Dinophysis, and the Mesodynium responds to temperature, if I remember correctly. Did the temperature influence the response of the Mesodynium to the Dinophysis?
That's a great question, and we're actually still trying to figure that out. So I'll go back to that graph. So we were trying to figure out why Mesodynium had this negative relationship with temperature and also what the relationship was. And interestingly, there's a couple different hypotheses of what's going on here. One is that because Dinophysis blooms in March, maybe when Dinophysis in the predator decreases, that allows Mesodynium the prey to increase again. So when the predator's present, it might influence how much prey is in the water. Another potential hypothesis is like what you were saying, is because Mesodynium prefers those colder temperatures. That's why we see a lot of Mesodynium, but no Dinophysis. So temperature and prey availability and that whole interaction between Dinophysis and temperature, they're all kind of working together, and we're still trying to figure out exactly what we're seeing. But yeah, there's definitely a couple hypotheses regarding that.
We have another question from the audience.
All right, Maya, great presentation. So I have two questions, but they're similarly related in your methodology, especially since you're using NOAA temperature data. So my first question is, with your sampling method using the IFC, so are all of your water samples or your image sampling just coming from off the VIMS dock, or are you also collecting water samples? Because one, like if you're taking it just from that one location, then you're not really getting a full idea of what the whole York River is showing as far as the Mesodynium and the Dinophysis. It's a snapshot to that one location. And then also, so how do you account for that one, like the spatial variability? And then two, similarly related with your NOAA data, like for instance, on this graph or this slide, the graph says Chesapeake Bay temperature data. So are you actually correlate or getting temperature data at the VIM stock, or are you using an outside area where the NOAA data is coming from? So is there a mismatch in the temperature to location?
Yeah, that's a great question. So the NOAA temperature data does come from the York River, and the IFCB is only collecting off the VIMS pier. And that's actually for a number of reasons relating to just the complexity of the IFCB. It does need to be pulled out of the water every so often in order to change the, we have chemicals in there to make sure that no organisms grow, and we have to monitor it. And there's a lot of factors that if it gets too high, it will stop collecting data or break. And it's a pretty novel instrument. So I think we're still on the early ends of figuring out how we can use it to best form data collection. But as more labs start to use it, because currently not that many labs have an IFCB, but as more labs start to use it, it would be very valuable to deploy it in other locations and see what we can see with trends and patterns.
All right, we have time for, let's see. We have a question from the audience, and depending on that, we'll see if we have time for one more question.
Really nice presentation, and I think that this work is a great next step to try to explore these things that have only been looked at in the lab, in the field. So this is really awesome to see. My question was related to something you said in the introduction, which was that these predator-prey interactions are generally not accepted in the field. And so although you didn't see the sort of blooming patterns that you expected, is there anything in the data that you did collect that could help inform this predator-prey relationship? Does it exist? What do you think?
So yeah, it was actually really interesting that we saw the patterns that we did with mesodynium peaking after. And kind of what I was saying earlier with potentially if Dinophysis being really abundant in the water, and then when it disappears in lower temperatures, that allows mesodynium to grow more, that could potentially relate to that predator-prey interaction. Other ciliates and prey groups have been studied in other areas, and especially in areas where Dinophysis is in higher cell abundance. Because it's in such small cell abundance in the Chesapeake Bay, it's really hard to observe these predator-prey interactions. And like what Stephanie had asked, we're only looking at that one location. So our data didn't show the trend you would expect for a predator-prey relationship, but we do know that mesodynium is a key prey group just based on what we've seen in culturing contexts. Does that answer your question? Okay.
All right, thank you so much, Maya. If we could get a round of applause. We don't have time for another question, but well, no, I don't think it's a quick question, but we can introduce the next person, and I'm really sorry to the person on Zoom. All right, so our next student presenter is Alexis Putney. She attends Old Dominion University, and her mentors are Dr. Emily B. Rivest, Annie Schatz, and Abby Sisti.
Thank you for that introduction. I'm so excited to show my research this summer. But my research was on diet diversity effects on the growth of the stressed eastern oyster larvae. And as Nihal said, I worked with Dr. Emily B. Rivest, Annie Schatz, and Abby Sisti. So to start off, why do we care about oysters? The eastern oyster, it's found in various locations, but it's also found in the Chesapeake Bay estuary, which is close to where we live. And it's also considered a keystone species. So keystone species is dependent on highly by other species. For example, the blue crab and the striped bass fish, which is found in our waters. And it's dependent on like for a shelter or for food. And they're also filter feeders, so they remove algae in the water. So here in this bottom picture, you can see there's two different tanks. And in this tank, the water is murky. So without the oysters, algae will grow close to the surface of the water and sunlight isn't able to go through. And yeah, like I said, it just makes the water murky. As to where in this tank, you can see that with the oysters, they'll filter feed the algae and clean the water. But for my project, I specifically looked at oyster larvae, and they are more vulnerable and they need more energy. And I say that because an analogy that my graduate student gave me was, think of us as babies. We need energy to grow, we need food to grow. So I'm also looking at stress oyster larvae. So climate change may negatively impact oysters. Environmental stress is an example. It's when there's a change in the temperature, salinity, or the nutrient of the water, and it can affect the marine organisms by messing with their growth. Another example is ocean acidification, and that's when there's a decrease in the pH, and it will cause the water to be more acidic. So in this picture here, you can see that right here, the pH is dropping. And shellfish, such as an oyster, they use calcium and carbonate ions to create their shells to make it strong. So with the pH dropping, it will cause the water to be acidic, and there will be fewer carbonate ions for them to make their shells, like I said, with calcium. So the shells will be weak, and they'll be smaller, and there'll be fewer amount of shellfish. And like I said, it can stunt their growth. And with them being stressed, they're going to have to use more energy. And with using more energy, I'm also going to look at their diet. So correct diet is important for oysters. The oysters I got are, they're not from a hatchery, they're actually from the Rappahannock River, but sometimes oysters come from a hatchery, and a standard hatchery has a diet. And with the diet, in the early stages of life, they're fed only one algal species that has a lower density. So if they're smaller, they're going to be fed a smaller amount of density in, it's going to be a smaller density that they're fed. And then later stages in their larval life, you start to add more amount of algal species. So those algal species will have a higher density. And in the third bullet, you could see larger algae is introduced as the larvae will grow. And an example I got from my graduate student, Annie Schatz, was in 2019, she had conducted a project. And in her project, she only fed her larvae one algal species. And by doing that, the larvae didn't make it to metamorphosis, and the cultures had crashed. So hearing that, I want to look at, if I feed them more than one diet, how will that work? And so diet is a source of their energy. And like I said, they're going to be dealing with stress and the growth. So how much is the diet going to incorporate in their growth? So my research question and hypothesis is, how will larval growth be affected by diet diversity? If I take a group of stress oyster larvae and feed them a diverse diet, they will be larger in size. And how I'm going to measure their size is looking at the area and length of their shells. So I had multiple methods in my project, and for my first method to start off the project, I got adult oysters from the Rappahannock, and we did a spawning process. And then after the spawning process, we got our stress oyster larvae, and they came from a high temperature of 29 degrees Celsius and a low pH of 7.2. Then after 6 days, I created 2 treatment groups. And here you can see I did 2 different containers. So my one container that is pink is my control group, and they were fed only one diet of algal species. And my second container that is in red is my diverse diet group, and they were fed 2 types of algal species. And then after putting the larvae into their treatment groups, I would feed them every day. And both groups always got the same cell density, and I would pipette it into the containers. And then here's 2 pictures. This is where the oysters lived in the tank room of our lab. And I would put them, as I said, into their containers, and then put the containers into the tank that were filled with treatment water. So my second method was my water changes. So my experiment ran for 5 days, which means the larvae were 11 days old. And every 2 days, I would change the water. So starting that, I would get a new container and fill it with new treatment water. And then I would pour the water into a 55 micron sieve. And after pouring it into the sieve, the larvae would be caught onto the mesh. And then I would pour the remainder oyster larvae from the mesh into the new container and rinse them in there. And then my last method was for my data analysis. After my experiment of 11 days, I would collect the samples, and I had to make sure that they were rested at the bottom so I could take my pictures. And after they were rested, I would put them onto a slide and look at them under the microscope. And then looking at them under the microscope, this is what I could see. So here you see, this is what it would come up from the camera on the microscope, and it would show the slide where the larvae are at. And so I would have to take multiple pictures for each treatment group and had to make sure that I had at least 50 good larvae to measure. 50 is just a number that we use in the lab, but I had to make sure that I had at least 50, that they weren't dead, and they didn't have any extra stuff on their shell because it would be included in their measurements, and that wouldn't be fair. And so then after taking my pictures, I would analyze my images, and I did it through a software called ImageJ, and it would calculate the area and length of these shells for me. So in my results, this is my graph. And so on my x-axis, I have my treatment groups, which is my control in orange and my diverse diet in blue. And on my y-axis, I have my average of the area. And so looking at the graph, you can see that the diverse diet group were higher in the area than the control group. And when conducting for my significant analysis, I did a statistical analysis, I did a t-test, and for my t-test, my p-value was higher than 0.05. So with that being said, if you look at with the statistical analysis, it says that there's no significant difference between the two groups. But also, I did standard error bars, which are these that you can see, and they basically show the consistency of the growth between the treatment groups. So with the long bar, you can see that it's showing that there was a variety in sizes, and then with a shorter bar, it's saying it was more consistent in being higher in growth. So again, and also this is a picture of what I measured. This is what the area was of the oyster shells. And then here's the length that I measured. And then again, you can see that the diverse diet was larger, and my t-test, the p-value was above 0.05, and my standard error bars show that it was more significant in the diverse diet than the control group of the size of the oyster larvae. And then this is my graph of my length, sorry. So to conduct my research, a diverse diet may help larvae in the future. So with high temperatures and low pH conditions, the stressed larvae, a diverse diet group were greater in area and length. And as I said, there was no significant difference with my statistical analysis, but with the standard error bars, you can see that larval growth was still more consistent under a diverse diet. And with this data, I can tell a hatchery it's more beneficial for culturing in the hatchery, and they can understand that if they continue to feed larvae a diverse diet, they'll still be greater in growth. So it could benefit the hatcheries by not making their business come to a stop and making sure everyone still has a job because they're still taking care of all these oysters. And it also shows them with climate change that if they're dealing with oysters that are stressed coming from all these different environments, if they feed them a diet that is diverse, it will be consistent in a larger growth. And then for future studies, I could test with more individuals. So in these two pictures, these are pictures of juvenile oysters. These are older than the oysters that I used. So I could test with multiple or more individuals. So I could have a bigger, larger group of the amount of oysters I use, or I could test it for a longer period of time with different age groups. As I said here, these are juveniles, so it might be different in how much they eat. And I can also test with different diets. So for my project, as I said, I only use one. My control was just one diet, and my diverse diet was 2 species. So I could test with different diets and maybe do one group getting 3 types of species and another group getting 4 types of algal species. And I would like to acknowledge my mentors, Dr. Emily B. Rivest, Annie Schatz, Abby Sisti, and the Rivest lab members, especially Brett and our REU coordinators and all the funding that we received to conduct these projects. And now I'll take any questions.
All right, thank you, Alexis. So we will be taking any questions from the audience. I see one from Dr. Seitz right now.
Hello, great job.
Thank you.
Nice study. So my question is, both of your treatment groups were with stressed larvae, and I'm just wondering if you used a treatment group that wasn't stressed, how you think they would fare with the different diets?
I feel like even if I used a group that wasn't stressed, the results would probably still be the same, just because like the analogy I use, like if you're younger, you're going to want to eat more food. So with the diverse diet, if they're getting fed 2 types of algal species, they're going to grow larger than a group that's only fed 1 algal species.
So your recommendations for hatcheries could apply even if they're not experiencing, really low pH values and so on.
Yeah, and then also Annie's project that she's working on now, she's working with a whole bunch of multiple stressors. So it's interesting too to see how like a change in the temperature, a change in the salinity, like the different growth that they have, even though she's not looking at the feeding, just the different growth that they even have just on the water.
Great, thank you.
All right, I believe I saw another question from the audience from Amanda.
Hello, nice job.
Thank you.
So you mentioned the diverse diet, right? So are there benefits to using different types of algae to feed the oysters in general? Like if you fed one type, does it have a benefit this way? Yeah, there.
Yeah, so if you remember when we went to the hatchery, oops, I forgot her name, but she was telling us like the different, if you look at this picture here that I took, you can see that there's multiple types of algal species, and each algal species has different nutrients that it comes with. Now, when they usually start the diet, they always feed with pavlova, as she said, which looking at the words that it says, they just all come with different nutrients. So the hatchery, she was just telling us that they start off with pavlova and then they grow from there.
All right, I also saw a question from the audience from Duncan, and it's covered? All right, sounds good. Oh, but we do have one from Stephanie, so go ahead.
Hey, Alexis, great job.
Thank you.
My question is just more general. So is your research for the summer, is it geared more towards understanding how these oysters are going to behave in nature or the wild? Or is your research directed more at like fisheries and aquaculture?
My research was more so towards aquaculture and hatcheries, because like I said, I was looking at specifically stress oyster larvae. So I want to look at like how ocean acidification affects, how environmental stress affects. So with those factors, they're more so used in a aquaculture hatchery. So that's basically what my research was just looking at stress oyster larvae and how their diverse diet will affect them.
Thank you.
All right, is there any other questions from the audience or Zoom before we move on to the next person? All right, we do have a question in the audience. So one more for you, Alexis.
Want to make sure we use up the full time slot. Great job, Alexis.
Thank you.
So if the REU program were extended another 2 months, what would be the next experiment you'd want to do?
With my specific project or anything?
Anything.
Oh, Well, with my specific project, I think I would want to look at an older age group, because if you guys didn't know, but I was supposed to conduct the experiment twice, and I was doing it with first like 6-day-old larvae, and then I was going to do it again with the 11th day old ones, but it ended up crashing and they all died. But anyway, so I would try to do it again and maybe do it with older oysters. And if I wanted to do anything, I think I want to work with like some sea turtles or something.
All right, well, good job, Alexis. Thank you for answering all those questions. For our next presenter, he will be joining us through Zoom virtually. And so I don't believe you will see him presenting because he is going to be on the computer, but his name is Galvan-Dubois, and he attends Cornell University up in New York, and his mentor is Dr. Andrew Wargo. So Kai, if you can hear us, you can say hi.
Hey, can you see my screen?
Not yet, Kai. Just give us a moment of technical adjustment as we're switching from a different type of shared screen. Okay, Kai. We can see your screen and we can see you, and as soon as you start presenting.
Okay. So my REU project was on the effect of rainbow trout genetic variation on the susceptibility to viral disease and infection. I'm Kai. I'm from Cornell University, and my mentor was Dr. Andrew Wargo, and we were studying the Department of Aquatic Health Sciences. So the host for the pathogen that we studied was the rainbow trout, and it's a salmonid endemic to the Pacific Northwest of North America. And this fish is a good aquaculture species because of the fact that it provides protein and can be easily distributed worldwide in different forms, such as filleted or smoked. Also, another main reason why it's very good for aquaculture production is because of the fact that it's very adaptable to life in domestication and easily can be grown. So this leads it to be popular aquaculture species worldwide, which makes it a very economically important species. So one of the main problems with aquaculture of the species, though, is pathogens, and they can greatly hinder the production. So the pathogen that we're studying was IHNV, or infectious hematopoietic necrosis virus. It's a rhabdovirus that causes the necrosis of interior kidneys, basically just leading to the kidneys of the fish completely failing and shutting down. So clinical signs for this pathogen include darkened body, lethargy, pale gills, bilateral exophthalmia, and distension of the abdomen, hemorrhages at the base of fins. So in the image, we can see a non-infected fish and then an infected fish, where the infected fish is much darker and has eyes that are basically popping out on the sides. And then towards the body is way more swollen as well, where the fins are. And so, yeah, this pathogen is a major problem for aquaculture, both aquaculture and wildlife. And in the production of aquaculture can result in up to 90% of fish being lost. So it really increases the management costs by making people usually having to be more strict with their aquaculture in the form of quarantining, or when the infection happens, they have to foul their systems. So there's additional costs from that. And so therefore, previous research has mainly been done on selective breeding for disease resistance against these pathogens. But for my project, we looked more at the relationship between resistance to disease and resistance to infection, and whether these traits would be, whether it'd be possible for these traits to be selected for the breeding programs. Because the main difference is that disease resistance can prevent mortality, but does not actually prevent infection all the time. So that would still lead to basically asymptomatic carriers, and because of the reason that disease resistance basically just blocks the symptoms and decreases viral load typically. So that doesn't actually reduce asymptomatic carriers. And so the disease is still able to infect hosts and co-evolve to become more virulent or more dangerous. And so with infection resistance, though, that would be able to prevent both asymptomatic carriers and the disease from becoming more dangerous. So that would definitely help with eradicating disease. So the previous work done, there were 2 experiments conducted independent of each other, and the traits were assessed. So infection resistance and disease resistance in different trout families, with trout families just being fish that came from the same parents, basically. And so in the first study conducted at VIMS, we had 20 different trout families, and they were all given multiple doses of IH&V to create dose response curves. And there was mortality of triplicate groups of 20 fish per family recorded. So basically the amount of fish that were dying. And then also the shedding of individual fish was also recorded using quantitative PCR. So these fish were kept in individual tanks. And then the second study in Rivest was another aquaculture facility with 82 different trout families. And they were only given a single dose of IH&V though. And the shedding and mortality of 2 groups of 50 fish per family were recorded. So basically these statistics for the second study were recorded for the tank rather than the individual fish. And so basically we have the fish, we infect them with IH&V, and then for the disease resistance part, we just count how many fish die and how many survive through the experiment. For the infection part, we give them the disease, and then we take water samples from the tanks they're in, and we run it through quantitative PCR to see how much viral load there is. So we hypothesized that for the first hypothesis, rainbow trout families do have a significant difference in their survival. And rainbow trout families differ in the amount of viral shedding and the number of fish shedding. And then our last hypothesis, that there would be a relationship between these 2 traits. So basically a relationship between disease and infection and transmission resistance. So the data that was collected for the VIMS study was daily survival was recorded as a binary variable. If it died, a 1 was marked. If it survived, a 0 was marked. And so the amount of virus that was shed was also quantified from the individual fish on the second day of the experiment in order to get the most amount of shedding recorded. And family and the virus exposure dose were used as explanatory variables in our models. And the tank random effect was also recorded, just like the tank that they're kept in was also recorded just in case they need to be implemented as random effects in our models. And then so for the Rivest study, we recorded the total number of fish surviving, the amount of virus shed recorded from group of fish tanks on the third day for, again, the maximum amount of shedding during the experiment, and family was an explanatory variable. But again, we only used 1 exposure dose. And then the tanks that they're kept in was also recorded in case it needed to be used as a random effect. So the methodology and data analysis part of my project for quantifying the susceptibility to disease and infection was, for the disease part, the models that we used were Kaplan-Meier survival curves, Cox proportional hazard models, and logistic regression. And then for the infection resistance part, we used the proportion of fish that were shedding to create a logistic regression curve. And then we also used the amount of virus that the fish were shedding just to make a general linear model. And we also calculated ID50 values from logistic regression models that were made for each individual fish's family. And basically ID50 values are just the amount of virus exposure dose required to infect half of that fish's family. So for the third hypothesis, the correlation between disease and infection, we used family as just a covariate and ended up using a quasi-binomial regression in the Rivest study in order to see the relationship between disease and infection. And all the data analysis was conducted in R and Excel, with mostly being in R. Excel was just used for simple plotting and linear models. And we chose the model selection process was largely based on choosing models that we're interested in that had lower AIC and better likelihood if they were nested models. So we just wanted to maintain parsimony by selecting the simpler models. A literature review is also conducted with search terms including selective breeding and infection in fish and mortality and infection levels, with the search engines mainly being Google Scholar and Scopus. So for the results of our disease resistance, we have in the VIMS experiment, we have on the x-axis the days of the experiment, and the y-axis we have the proportion of fish surviving, with each of these curves representing a different family. So the colors correspond to each of the different families. And we found that families 5, 6, 9, 10, and 12 all had a statistically significant greater survival than the dark black line, which was basically just a curve, another Cox proportion, a Kaplan-Meier survival curve that did not separate the family. So that was just all of them combined into one curve. So we see 6, 9, 5, 10, and 12 all had higher survival using 95% confidence intervals. So those bands are just 95% confidence intervals. So for the next results, we have on the x-axis, we have the virus exposure dose, and on the y-axis, we have the log quantity of viral RNA being shed by the fish from qPCR. And so here, there was actually a statistically significant interaction between the family of the fish and the virus exposure dose that they were given. So basically that just means when, as virus exposure dose increases, so did the viral shedding increase, but the amount it increased by differed between each family. So basically we just had different slopes between the different families. And we compared, for this analysis, we used the baseline family was set as family 16 just because that had the highest amount of shedding. And so when we compare these curves to family 16, which is the red curve at the top, we can see that families 8, 6, and 9 all had, they all had a statistically significant, less of an increase in shedding than family 16 for each different virus dose. Okay. Next results was on the x-axis again, virus exposure dose, but this time on the y-axis we had percent infected, and that was just approximated by the percentage of fish that were shedding, because if fish are shedding, we can assume that they are infected by the virus. And again, we compared the different curves to the family 16, and we can see that actually in this analysis that all of the different families had appeared to increase more slowly as a function of dose for every family compared to family 16, because of, again, the interaction between virus exposure dose and family. And so the bands, again, are just 95% confidence intervals, and you can see they increase more slowly compared to family 16, which increases very abruptly in the amount of fish that are being infected at each virus dosage. So, and then for the relationship part of the experiment between disease resistance and infection resistance, we plotted the proportion dying against those ID50 values I was talking about earlier, and we found that 66.94% of the variation in mortality was explained by those ID50 values, and each point is basically just a family, and so that's a pretty strong correlation we have here, and it's a negative relationship, which makes sense when you think about it, because as the virus dosage increases, as the more virus dose that you require to infect half of a family increases, so should the proportion that are dying decrease, because that would mean that they are more resistant. And so again, now so basically same relationship, but different data now. We're using the Rivest experiment, and we have on the y-axis the quantity shed, and we just log transformed it. And on the x-axis this time we have the proportion mortality. And we could see that families differ in mortality and shedding, and that there was a correlation, but it's much weaker this time, with only 15.25% of the variation in log quantity shed being explained by the proportion of mortality for each family. And yeah, so not all the variation in mortality is explained by shedding clearly. And for example, when you look at family 13, even though it has the least amount of shedding, it does not have the least amount of proportion, and it's still like towards the middle. So the conclusions we have are that trout families differ in their survival and transmission, and that survival and transmission appear to be linked at the family level. So this would indicate that these traits could be heritable, but we also saw from those last two plots that disease resistance did not always provide infection resistance, which is helpful when you're trying to determine selective breeding programs, because now you know that you can't just select for one trait and get the other. You'd have to specifically choose which trait you want and prioritize, and yeah, which trait is more important to you. And you can also try to identify fish with both traits. So aquaculture should definitely consider the importance of both of the traits for selective breeding. And then what's next is that this study basically laid a framework for future research on selective breeding for infection blocking, whereas in the past it's mostly been on disease resistance. So, and the next thing we could do would be to determine the heritability of the disease resistance and infection resistance traits. So more on the genetic side of analysis, because this was only at the family level. And we could also increase the study size and increasing the replicate and sample sizes, because, for example, in the Rivest study, those were only duplicate tanks, which made it very hard to use general linear models with just such a small replicate size. So that's why we usually didn't do that. And yeah, we could also identify the genetic determinants of infection resistance. Basically, we could do more analysis on genetics and try to map these genes and figure out if they're linked. So I would just like to thank the Wargo Lab, VIMS, NSF, REU, Rivest, USDA, and USGS. And I could take any questions now.
All right, thank you, Kai, for that presentation. So we'll take any questions from either the audience or Zoom. And so we can start now. Oh, we do have one from the audience.
Great job, Kai. It was really interesting to see all of your results. I was wondering if you could go back to one of the slides of the VIMS results. And I noticed that the disease load, percent infected, differs among the families with 0 virus exposure dose. And so I'm wondering if that means that there's some sort of like background viral load, and if you think that had an effect on the results.
Yeah, so I think that's mainly just from the way that the model was just constructed, that the percent infected, we still got percent infected just because it's approximating based on all these different dosages. So that's mainly the reason why, because usually, or actually I think the main thing was that because we were only using percent of fish that were being infected, so we didn't always include, or actually, I'm actually not quite sure why we got higher than 0 values. But yeah, that's a good question. I'm sorry, I don't have the answer.
All right, thank you for trying to answer that question. And it looks like we have a question in the chat. And they ask, are the families that have higher survival on your survival curve bred for INHV resistance?
Oh yeah, so that's a good question. We did actually have so basically the first 10 strains were supposed to be resistant strains, and then the latter 10 strains were supposed to be more susceptible strains. So yeah, that's how that was.
All right. We have a question from the audience from Duncan.
Hi, Kai. Great presentation. So I was a little unclear. The virus you're studying, is it a single strain or is it a family of viruses?
Oh, it's just, yeah, it's just a single strain, IH and V. We weren't studying different strains of virus.
And is there, do you have to make sure that your population doesn't have any other viruses circulating? Can you get interference from other viral strains similar to the IH and V?
Yeah, that's why we had the 0 dose as well, so that we would also record the amount of viral load that the fish had at 0. So it wasn't many fish. There was actually fish that did have viral load at 0 dosage, but I don't think that it was too many.
All right, thank you.
All right, does anybody else have a question from the audience or Zoom? Okay, I have a question, if that's okay. It's okay if you don't know how to answer it either, but in regards, this is more like your introduction, just kind of implications of your study. There is usually, well, I've heard that there's a lot of problems when it comes to aquaculture for the rainbow trout because it does spread a lot of diseases, a lot of infection that spreads to the wild population. So do you know anything about that in regards to if this study, if they could control it in a wild environment, even though it is still aquaculture, but when they, like, it spreads to like salmon, for example, would that have the same implications or is it very species specific?
Yeah, no, it's a major problem because of the fact that it can infect different fish species as well. So definitely salmon would be able to get infected by IH and V as well.
All right, well, thank you, Kai, for the presentation. If everyone give a round of applause. And now we are going to introduce our last presenter for this first session. Her name is Amanda Salmaraji. She attends Purchase College, and her mentors are Dr. Jan McDowell and Ellen Bisak.
Hello, everyone. My name is Amanda Samaraji, and I've been working with Dr. Jan McDowell and Ellen Bisak, to determine relatedness of juvenile Atlantic striped bass within the Chesapeake Bay by using microsatellite markers. So the Atlantic striped bass is an anadromous fish, meaning it spends most of its adult life in the oceans or in estuaries, but then it comes back into rivers to spawn. So they can be found all along the east coast of North America, as far north as Canada, as south as Florida, and they're also found in the Gulf of Mexico. Striped bass are an important economic stock for the United States. In 2019, there were 14 million pounds of striped bass harvested by commercial fisheries, which roughly translate to $15 million for the United States. Recreational fisheries harvested even more than this at 23 million pounds. So in the late 1970s, due to an increase of fishing pressure and habitat loss, the stock of Atlantic striped bass collapsed. So this also led to the development of a fisheries management plan in 1981. However, as of June 2021, the striped bass is still overfished and it's still experiencing overfishing. Recently, genetic advancements have made investigations into population size, structure, and diversity much easier. So calculations of and estimates of relatedness, or how closely related two individuals are, have been used to investigate population diversity. One genetic marker that can be used are microsatellites. So microsatellites are the short tandem repeats of 1 to 6 nucleotides found throughout the genome. And as you can see up here in the picture, we can see these microsatellites. So they're the repeats, and for a dinucleotide repeat, you see here C and A, and then that would be repeated for n number of times. So for this example, C and A is repeated 8 different times within the genome. Microsatellites are used for many reasons. They, as I said, are distributed throughout the genome, are specific to a site on a chromosome, they're highly polymorphic, and they can be amplified through a polymerase chain reaction, so PCR. They're also codominant, so both alleles can be visualized. Microsatellites can also be used to calculate realized relatedness instead of expected relatedness that traditional pedigree calculations provided. If you have a sibling that shares the same mom and the same dad, you're about 50% related. By utilizing the number of fish by utilizing relatedness, the number of fish using and/or returning to spawning grounds can be identified, and eventually estimates of population size could be estimated. So overall, by furthering our understanding of striped bass genetic population structure within the Chesapeake Bay, we can help to rebuild the stock and increase abundance, stability, and recruitment. So this study has multiple hypotheses. For my project, I wanted to investigate if the broader questions could be answered using a smaller scale investigation. So we wanted to make sure that this was feasible before we launched a larger scale investigation. So to do this, I had to see if we'd be able to recover any full siblings in a small sample set from a tributary of Chesapeake Bay. In this case, we used the Rappahannock River, and my first null hypothesis demonstrates this. So it states that I will find no sibling or half-sibling pairs among the young of year striped bass sampled within the Rappahannock River of Chesapeake Bay. So the slash here just indicates that there's going to be no relatedness, and RAJ just stands for Rappahannock juvenile. The rest of my hypotheses deal with average relatedness among the juvenile pairs, and they were grouped together using 2 different criteria. So my second null hypothesis is that average relatedness levels will not differ among pairs of individuals that were sampled in the same month and those sampled in different months. So here the criteria is collection month. This is saying that average relatedness is not going to be significantly different depending on when the samples were collected. The second criteria that we looked at was size. So my third null hypothesis is that average relatedness will not differ among pairs of individuals that are in the same size category than are in different size categories. So in other words, it doesn't matter what size the fish is, we're not going to find a significantly different amount of relatedness depending on the size of the fish. So to investigate these hypotheses, I'm building off a prior study conducted by Michelson 2018. Here, she conducted microsatellite genotyping of 555 young of year striped bass from the Rappahannock River in summer 2016. So here is a map that shows just the Rappahannock River and the different juvenile sample sites that she took from. So these blue triangles show all of the sampling sites. Fin clips or muscle tissue were taken from the juveniles and were then used for DNA isolation. So Michelson used 20 loci in her investigation, and these loci were amplified for all samples using PCR, and that reaction also included locus-specific fluorescent labels. These labels would allow for multiplexing of multiple microsatellites in each reaction. So in her master's thesis, it was noted that some of the loci should be re-examined. So I used her existing data and recalled the alleles for 16 of the 20 loci that were deemed good for use. So I used GeneMarker, which is a computer program to evaluate chromatic peaks generated by a genetic sequencer. GeneMarker is a computer program that helps us visualize these sizes of the alleles as peaks of fluorescence. So this is just a screenshot from the program, so you can get an idea of what I'm looking at, and this is also a great example of what we want to see in GeneMarker. Keep this in mind. Each window here is an individual striped bass juvenile, and each peak that you see is for a different allele. Just as a reminder, an allele is a variant form of a gene, so remember Gregor Mendel and his peas, this is what we're looking at. It's a genetic trait. So after rescoring the data with gene marker, we noticed that there were some issues. There was low amplification with high background noise. There were multiple peaks, and there was potentially some cross-contamination that was going on. The presence of these issues made it difficult to determine what was real in the samples and what was not. In these cases, the original allele calls were deleted from the data and were redone. Because of this, 230 samples were chosen to undergo a second round of PCR to create a more accurate data set. So of the 555 samples that were collected in the Michelson study, 230 of the samples had to undergo the second round of PCR. And after PCR, these samples were then reanalyzed. Excuse me. After PCR, the samples were then transferred to an ABI 3500 genetic analyzer for sequencing. And we used capillary electrophoresis fragment analysis, which separates the fragments by size and measures fluorescence. This allows for the visualization in the program like GeneMarker. So a total of 193 individuals were removed from the data set due to missing greater than or equal to 25% of the allelic data that we're looking for across all of these 16 loci. This left 362 juvenile individuals that can be used for statistical analyses. So the programs that I used were MicroChecker, and MicroChecker is used to check for any scoring errors that happen and identify any potential for null alleles within that loci. So a null allele occurs when there is an allele that did not amplify due to a mutation, thus causing a greater number of homozygotes than expected in the population. Colony was used to evaluate full sibling and half sibling relationships and also to resolve family groups. We used an empirical data set analysis and conducted a medium length full likelihood run with high likelihood precision. We added a 5% allelic dropout error rate for any of the loci that held presence of null alleles, as was determined by MicroChecker. And a 0.5% genotyping error rate was added for all of the loci in order to account for potential scoring errors or PCR errors. We also opted to include inbreeding within the population. So the juvenile data was also run through co-ancestry to estimate relatedness between pairs of samples using the empirical data analysis option. It gives us our relatedness value, so the R coefficient that we're looking at, and the R value ranges from 0 to 1. So full siblings share about half of their genomic DNA, so the R value would be 0.5. Half siblings share about 25%, which would mean that the R value is 0.25. So co-ancestry used to calculate allele frequencies also, and we ended up using the triadic likelihood estimator. So Colony was able to determine that there are 9 full siblings and 562 half siblings amongst all of the pairs that we used. Full siblings are denoted here by the orange diamonds, and half siblings are shown by the green triangles. So of the 9 full sibling pairs, 5 of those pairs held a probability of greater than 90% for being full siblings. And for the half siblings, there were 93 pairs of the 562 that held a probability greater than 90%. So this indicates that there are full siblings and half siblings among the juveniles in the Rappahannock River. As a reminder, my null hypothesis was that we would find no relatedness, no half siblings or full siblings between any of the juveniles. So this is not supported by the data, and therefore we can reject it. However, the familiar relationships between the juveniles is not incredibly clear. While there is evidence for relatedness within the data, we are not able to determine a strict cutoff point as to where distinctions between full siblings, half siblings, and unrelated individuals can begin. So boundaries cannot be identified due to an overlap of data in these box and whisker plots. So you can see that the whiskers of the box plots are stretching into the other categories. These box plots were constructed using adult striped bass allele frequencies that was run in co-ancestry. So this issue can likely be remedied by the addition of more loci to the data set as well as more individuals. So this being said, there is a high possibility of the full sibling pairs being real. This is because the relationship threshold is much stricter than that of half siblings, and distinctions between full siblings and unrelated pairs is much clearer than between half siblings and unrelated pairs. So theoretically, full siblings share half, whereas half siblings share 25%. So because of the overwhelming number of half siblings, we chose to focus on the full sibling pairs. In this table, and I know it's a lot to look at right now, you can see the 9 pairs of full siblings from the previous image. So if you look at the R coefficient here provided by co-ancestry with the probability score given by colony, you are able to see that the 5 full sibling pairs here meet our criteria of having a R value of or around 0.5 and have a greater probability than 90%. So this indicates that the full sibling relationships here are real. And then if you bring your attention to the red outline here, you can see that there are 2 individuals with an R value of about 0.55 with almost 100% probability. So these juveniles were caught on the same day in the same sampling site and were about the same size. We wanted to further investigate these patterns of relatedness among these groups created by this metadata provided. This leads me to my other null hypotheses that look at average relatedness. So we separated all 362 samples by 2 criteria, and then we took the average relatedness values of these groups and compared them in a Welch's 2-sample t-test. We obtained average relatedness for pairs of samples caught in June, August, and then also in August and June. Here we found a significantly higher level of relatedness in the within June pairs, compared to within June and August and within August. This suggests that there is more relatedness at the beginning of the spawning season than later. This is probably because there are fewer fish in the river in the beginning than are later, and as more fish come into the river throughout the season, the level of relatedness will go down. There was also a significant amount of relatedness found in the larger juveniles that were sampled. We divided the length of the fish into 2 groups. So the small group ranged between 20 millimeters and 35 millimeters, and the large group ranged between 55 and 70 millimeters. So this also makes sense since the fish caught in the earlier months are now the larger fish, indicating more relatedness. So fish caught in the earlier season were more related due to fewer fish being in the population, which means that the larger fish being caught later means that the larger fish sampled in the later in the season should follow the same trend. You can see that the sizes on the x-axis and collection month on the y-axis on the scatter plot. So overall, we can identify that there is relatedness among the young of year striped bass within the Rappahannock River of Chesapeake Bay. Having conducted this study, we can see that there is potential for this investigation to succeed, but more genetic markers, more time, more samples will be needed in order to resolve these relationships. So this study does serve as a great first step into identifying relatedness. And by knowing the relatedness of these young of year individuals, we can eventually develop estimates of population structure and abundance. So these estimates can then contribute to the management of the stock of Atlantic striped bass. As I said previously, it is an important stock for fisheries for both commercial and recreational use. Thank you.
Thank you so much, Amanda, for the presentation. We'll now open it up to any questions that anyone has, whether, again, Zoom or in person. So feel free to ask away. All right, we have a question from Dr. Seitz.
Nice job, Amanda. So my question is, were these results what you expected? Because you gave us your null hypotheses, but I'm kind of wondering whether you thought the results that you found would have occurred.
Yes. So in the Michelson thesis, she found that there was relatedness in the adult striped bass that were being sampled. And the reason we were doing this is because there are conflicting, and there's conflicting opinions, I guess, in is there relatedness within the striped bass at the Chesapeake Bay. So we were expecting it based on her results, but we also want to make sure.
And just to follow up, were there other studies done in different rivers that showed similar results, or all the previous studies have been done in the Rappahannock as well?
That I will have to get back to you on because I'm not sure.
Thank you.
All right. Do we have any other questions from the audience? I know we're coming up on lunch, so everybody's probably hungry. Oh, okay. We do have one from Duncan.
A great presentation.
Thank you.
So I had a quick question about the last set of data you showed us, that plot with the larger fish. So if I'm interpreting this correctly, the conclusion there was that the relatedness increased because the population size decreased. The fish got
No.
Okay.
So we're looking here, we separated the fish into two size categories, and we're finding that there is more relatedness in the larger fish, which would make sense because if they're being sampled later in the season, they have time to grow. So if I actually go back here, you can see there's ones, this one, okay. So this one was sampled at 2 different times. So this is one individual 1, right? And individual 2. And this one was sampled earlier, and this one was sampled later. And you see how this one was 42 millimeters in size. And then when the second one caught later is 50 millimeters. So we see that there is the growth happening, which is why this makes sense as well. So these are the dates, and you see the growth just moving across. Does that answer your question?
I'm sorry, no.
Okay.
So to clarify, relatedness, we are talking about relation, like as in how closely related these two organisms are in terms of genetics, heritage.
Okay.
Right? Do I have the correct?
Yeah, so we were looking at average relatedness here.
Okay. Yes. So what is the explanatory variable for why would larger fish be more related compared to, why would the population of larger fish sampled also be more likely to be more related? I'm not fully understanding the connection between these two variables, besides just...
So I'm going to try to answer this. I'm sorry. So average relatedness when sampled in the earlier months was higher, right? So these fish are going to be smaller in those earlier months, but then they have time to grow. So if the smaller fish from the earlier months are more related, are then sampled again later, they're also going to be the larger fish. So that correlation, that trend of that group being sampled earlier have time to grow, and so now they're the larger fish.
Okay, so we're just looking at the same population.
Yes, it's the same samples.
Got it. Sorry.
Cool. Thank you.
That was a great way to explain that, Amanda. All right, well, if we could get a round of applause for a presentation. And I'm going to now hand the microphone back to Dr. Rochelle Seitz.
Thanks, Nihal. All right, well, thank you everyone for your attendance at our first session. And now we're gonna have a lunch break and we're gonna join back here at 1:30. So for those of you who can make it back, please come back then. And also to any of the mentors whose students presented this morning, we are also gonna have a reception around 3 if you wanna come back for that. And so before we break for lunch, just wanna give a final round of applause for all of our morning presenters. Excellent job. And thanks everyone in the audience. We'll see you back here at 1:30. Well, good afternoon, everyone. Welcome back from the lunch break for our afternoon session of the 2021 Research Experience for Undergraduates presentation of research results. And so, as I mentioned this morning, we're going to have one of our interns moderate the session and introduce each speaker. And we are also going to give each student a 20-minute slot for 15 minutes for their presentation and then another 5 minutes for questions. And so we'll accept questions from the audience. And also, if you're joining us on Zoom, if you can type your question into the chat and we'll read it out to the presenter. So for our second session, our chair of the session is Amanda Salmaraji. And so I'll turn it over to her. Here you go, Amanda.
Good afternoon, everyone. So our first presenter for our second session is going to be Sophia Cherico, and her mentor was Dr. Mark Brush, and she is from the College of William and Mary.
Thank you. Do I just pull it up already?
Hi, everyone. Thank you, Amanda, for the introduction. Like she said, my name is Sophia Cherico. My advisor was Dr. Mark Brush, and the research I did was on the carrying capacity of oysters and the competition between aquacultured bivalves in Cherrystone Inlet. So what I'm going to be talking about today is a little bit of background first, and then I'll tell you about the objectives of our project, and then I'll talk about how we set up our research, the modeling we did, and what we found. So aquaculture is a growing industry globally. In the Chesapeake Bay, there's a lot of aquaculture specifically around bivalves. Cherrystone Inlet, as you can see on the map here, is located on the Virginia eastern shore, just north of Cape Charles. And it's actually the epicenter for hard clam aquaculture. So that is where we decided to focus our research. And typically, clams are the most popular aquacultured species, but they are also beginning to aquaculture oysters there. So hard clams and oysters are functionally very similar. They are both native filter feeders in the Chesapeake Bay. Hard clams are grown under nets on the bottom, and the harvest time is between 2 and 3 years, and they are market size when they reach over 40 millimeters in size. Oysters are grown in bags in the water column or in cages in the bottom. They are also harvested after 2 to 3 years, and their market size is a little bit larger at 63 millimeters. So our study was looking at carrying capacity, and there are a bunch of different ways that you can define carrying capacity. What we wanted to focus on for this study was production, And so we wanted to look at the maximum number of each bivalve that could be grown, and we wanted to ensure that each of them was reaching a harvestable size. So how we went about doing our research was with a model. We modified a Kushner model that was made in 2015. Kushner made his model to look at hard clams and cherrystone. And the way that he constructed his model was that he divided cherrystone into 7 boxes. As you can see in the image, boxes 1 through 3 span the inlet. Boxes 2 and 3 are the areas where the aquaculture farms are located. So those are the areas that we focused on specifically. And you can also see in the chart that boxes 4 through 7 divided the watershed, but we didn't focus on those as much as there were no bivalves growing there. The type of model that he made was a mechanistic ecosystem model. So we have our input values minus our output values, and that gave us growth that was calculated in grams of dry weight. The input values we used were ingestion, and output values were respiration, egestion, and for clams, reproduction. So the objectives of our study were, number 1, to modify the Kushner model, 2, couple the oyster model to the existing hard clam model, 3, we wanted to define the carrying capacity of cultured oysters, and 4, to examine competition between clams and oysters. Our hypothesis was that the carrying capacity of oysters would be similar to that of clams because functionally they are so similar. And we hypothesized that the population of oysters would be limited in size by food availability, and that is what Kushner found for clams. And finally, we hypothesized that the carrying capacity of bivalves when they're grown together will increase. So the first thing we did was alter the Kushner model. When Kushner made his model, he took specific data from the years 2001 and 2002. And what we found when reviewing his data was that in 2002, there was a spike in total suspended stollids, which is also called TSS. And that was an unusual reading and measurement. And so to make our model more generalizable, we took average annual cycle data that was collected over 20 years and averaged. After we modified the model that Kushner made, we incorporated the eastern oyster model. And then once it was coupled and running properly, we did our oyster runs and simulated populations from 25 million to 500 million. And those are the same runs that Kushner did with clams. And then we did combined clam and oyster runs where we simulated both clams and oysters when they were grown together. So after we modified Kushner's model, we wanted to make sure that our output values for environmental conditions matched his, even though we were using average data. So you can see here, this is the model output that we got when we ran boxes 2 and 3 with 100 million clams, and we looked at the dissolved oxygen levels. So you can see in the graphs, both of them show the cycles and annual patterns that Kushner found, and they matched really, really well. The next thing we looked at was dissolved inorganic nitrogen, or DIN. And so what we were really focusing on was that the patterns and cycles were the same. And so you can see here, Kushner's output is in blue and mine is in red. And so with the exception of the end of 2002 year data that Kushner had, which was the spike, like I mentioned earlier, our data does match pretty well. And so we felt comfortable that it was producing good data that we could use. The next thing we looked at was dissolved inorganic phosphorus, or DIP. You can see here that our annual cycles do match up with Kushner's again. The real exception is the spike at the end of 2002. Finally, the last environmental condition that we looked at was phytoplankton. We're using chlorophyll measures as a proxy for phytoplankton. So our peaks match really well with Kushner's. And the peaks are in the summer months, and the summer months are the time when the growing happens at the fastest rate. During the rest of the year, however, we do overestimate phytoplankton and chlorophyll levels, and we acknowledge that because of this overestimation, our predictions of populations are a little bit higher, but overall phytoplankton is within the same factor between both of our data sets, and because during the times of year with the highest growing periods, because they are the same during that time, we felt comfortable using this data. However, as I continue to do research in the fall with this project, we would like to refine that data set a little bit more. So after we know that the environmental conditions matched well, we added clams to the model. And we reran all of the runs that Kushner did. Again, those started at a low density of 25 million all the way up to 500 million. So this graph here The graph on the left shows the clam growth projections in box 2, and the graph on the right shows box 3. The y-axis is the shell height that we were measuring, and the x-axis shows the days of model run. So it was run for 3 years. And you can see that our projections in red are a little bit higher than Kushner's projections in light blue and dark blue. And the reason for this is because of our overestimation of phytoplankton. But you can see that the changes in rate and the trends and patterns of the graphs are the same. So we look deeper into this data. And so on the left, we have Kushner's analysis of growth rate from 0 to 500 million clams. And on the right, we have my analysis of growth rate from the same population sizes. So you can see here that the trends of the lines and the patterns are the same between Kushner's data and my data. And so this shows that over time, as the population of clams increases, the growth rate decreases, which is what we would expect. The next thing we looked at was harvestable biomass. So we looked at, as you increase the number of clams in the x-axis, how does the harvestable biomass change? And you can see in both Kushner's graphs and my graphs, around 200 million, there is the inflection point of this graph, which highlights the diminishing returns that you're getting. And so because of these diminishing returns, both Kushner and I would agree that the carrying capacity for oysters is around 200 million. Next, we looked at days to harvest. If you'll remember at the beginning, I talked about how clams are harvested after about 3 years. So on the graph on the left, Kushner put in a red line that shows the 3-year mark. So the y-axis is days to harvest. And what we did and compared in these graphs was we wanted to make sure that at every population that we tested, that the clams were growing to a harvestable size within the 3 years. So as you can see in both graphs, at all of our population sizes, the clams are reaching that harvestable size. The last thing we looked at was phytoplankton. Kushner found that phytoplankton was the limiting factor for clams. So in his graph on the left, you can see that as the number of clams increases, the chlorophyll levels decrease. The red line that is in his graph shows the baseline for chlorophyll levels in the inlet. And so if you look at my graph on the right, you can see that as the number of clams increases, the chlorophyll decreases, just like we saw in Kushner's. And by the time you're reaching 500 million clams, you are still a little bit higher than the tidal baseline. Okay, so after we did all the analysis for hard clams, we felt confident that our model output was similar to Kushner's and it was generating good results that were valid and possible in cherrystone. And so we decided to add our oysters. So for our oyster runs, we did the same runs of 25 million to 500 million, and we found something very surprising. We found that none of our simulated oysters, even at 25 million, were growing to a harvestable size. So when we graphed our annual growth rate, we could see a similar trend in growth, that it was decreasing as the population went up. But even at 25 million, the growth rate was much lower than we expected. When we graphed the average phytoplankton levels, as you can see on the right, the phytoplankton levels, even at 25 million, are much lower than we expected. And the chlorophyll has already been drawn down to below the baseline of cherrystone inlet. So from this, we know that oysters are using all of the chlorophyll that they have accessible to them, which is probably why they're not reaching a harvestable size. Because of this, we decided to do new oyster runs. So we started runs at 10,000 individuals and went up to 25 million. And so the results from those runs are graphed below. So on the left, we have average annual growth rate, and you can see that the growth rate at 10,000 up to 1 million is much higher than it is at 25 million. And you can see on the graph on the right, the average phytoplankton, again, is much higher at lower densities, and by 50 million, it is back at that baseline level that we saw in the other runs that we did. So what this tells us is that oysters filter chlorophyll at a much faster rate than we expected, and cherrystone can support far fewer oysters than clams. So the next thing we did was look at full oyster runs because we still wanted to know how many oysters could be grown and harvested. And what we found was that when the population exceeded 10 million individuals, none of them were reaching a harvestable size. So if you look at this graph on the left, we are looking at time to harvest. So the y-axis is days and the x-axis is millions of oysters. The blue line is box 2 and the red line is box 3. And you'll see that in box 2, when the population exceeds 1 million, the harvestable biomass decreases to 0. So after that population exceeds 1 million, you're no longer getting oysters that are large enough to harvest. In box 3, the individuals are growing a little bit larger, but by the time you exceed 4 million oysters, again, you're not getting any that are reaching a harvestable size. The graph on the right looks at harvestable biomass, and we see similar trends of growing better in box 3 than box 2. In box 2 on the graph on the right, again, once you exceed 1 million individuals, the biomass drops to 0, so none of them are reaching a harvestable size, and so none of the biomass that is there is harvestable. And when you exceed 4 million oysters in box 3, the harvestable biomass decreases because you can't harvest any of those because they're too small. So with this information in mind, we were thinking about how we would examine oysters and clams together. We knew that our model output was giving good readings for clams and oysters separately, and we wanted to know if you grew them together, what your results would be. So a simulation that we ran was looking at growth rates at 200 million individuals. So we ran clams when they were grown alone at 200 million individuals. We did a simulation that was a total of 200 million individuals, 100 million clams, and 100 million oysters. And we ran a simulation that was 200 million oysters that were grown alone. So you can see here in the bar graph, the 2 bars on the left are clams, and the 2 bars on the right are oysters. The bar on the far left shows the growth rate, the average growth rate over 3 years of the 200 million clams when they're grown alone. The bar to the right of that is blue, shows the average growth rate of the 100 million clams when they're grown in conjunction with the 100 million oysters. The first red bar shows the growth rate of oysters when it's grown just 200 million oysters and no clams. And finally, the final bar on the right shows the growth rate of oysters when you have 100 million oysters and 100 million clams. And so what we know is that when oysters and clams are grown together, the growth rate of clams decreases, but the growth rate of oysters increases, and this is because the clams are competing for chlorophyll, and that is the limiting factor. So the conclusions from our research is that the carrying capacity for hard clams is greater than the carrying capacity for oysters. Both oysters and clam populations are limited by food availability. And finally, the carrying capacity when bivalves are grown together will not be the same as when clams and oysters are grown alone. Future research, as I said, I think it's important to refine the chlorophyll prediction and make it more accurate throughout the year. It would be good to do further research to investigate the interactive effects of clams and oysters. We also noticed that oysters grow better in box 3, and at lower densities, clams grow better in box 2. So it would be interesting to look at how spatial variation and distribution of aquaculture farms could increase carrying capacity overall. I want to thank Dr. Mark Brush, my advisor, the Brush Lab, all of the VIMS REU interns and advisors, and NSF. Thank you. Gladly take questions.
Thank you, Sophia. We will now take any questions. Oh, we have a question from the audience, so Nihal.
Okay, good job. I did have a quick question that you definitely mentioned in the beginning. I just forgot. What was the difference between box 2 and box 3 again?
So I can go back and, sorry, show you the picture. Sorry, it's back at the beginning. Okay, so this is a map of Cherrystone Inlet. So box 3 is closer to the mouth of the Chesapeake Bay, and box 2 is in the middle, a little bit further in. And those are the only 2 boxes where there are aquaculture farms. Sure.
We have another question from Dr. Seitz.
So just following up on that, you're showing us where they are physically, but what differs between those two boxes and why might you get different growth rates in the two?
So we would want to do more research to look at the differences between them. We know that the limiting factor is food availability, and the food comes from the tides that's washing it in. So we would have to do more research, but I would hypothesize that maybe that there is more phytoplankton in box 3 that's available for them to use, but I'm not exactly sure.
Great, thanks.
I'm going to jump in and ask a follow-up question to that. So with boxes 2 and 3, do you know if there's a salinity gradient amongst those two boxes that would impact the growth rates?
Yes, so salinity in cherrystone does not vary all that much between box 3 and box 1, all the way up at the top. So I don't think it would be enough to impact it. If there is a difference, it's very, very small.
We have time for one more question. So, oh, Dr. Honig in the back.
Just a comment, you had a graph where you had on the y-axis days to harvest and on the x-axis the abundance. And you said if the abundance that you put in was too high, then you had the graph go down to 0. But really the graph should be going up to infinity, right? Because it's not that you can harvest after 0 days. That would be wonderful. And if you interpolate, it's basically saying the more that you still can get yield. But really what you're saying is if there's too much, you can't harvest at all. So you'd have to wait till the cows came home in order to be able to harvest. So it should be shooting up to infinity.
The reason that we put it down to 0 was because when the population exceeds a certain number, the individuals aren't reaching a harvestable size. And so that would make the harvestable amount 0, even though there is still biomass of individuals. They're not at a large enough size to take
Right,
them.
but your graph wasn't showing the biomass, it was showing the time to harvest, and the time to harvest is infinity. You'll never get there. They just won't get legal size. Because if you interpolate between that 0 point and the previous point, it would imply that halfway in between you're getting half the yield. And it's like, no, at a certain point it's going up to infinity. You're just not getting them big enough.
That makes sense.
Okay, thank you, Sophia. So we are now going to move on to our next presenter. All right, so our next presenter is going to be Annabelle Gorman. She's from Gettysburg College, and her mentors were Dr. John Honig and Julie Gross.
Thank you for that introduction, Amanda. So like she said, my name is Annabelle Gorman. I attend Gettysburg College. My mentors this summer were Dr. John Honig and Julie Gross. And this summer, I analyzed if there is an association between poor recruitment and environmental variables, more specifically, if extreme conditions can predict poor recruitment. Okay, to start with a little background information, recruitment is the addition of new juveniles into a population. This usually occurs at the age when they are first able to be fished. This may vary across different species and stocks. For example, one stock of cod could be recruited at age 1, while others might be recruited at age 2 or even 3. Additionally, recruitment is highly variable between year classes. Some years will have very poor recruitment and other years will have extremely high recruitment. And it's important to note that these really great years can be 10 or even 100 times greater than the really poor year classes. An example of this disparity is the so-called gadoid outburst of the 1960s. During this time, gadoids like cod were suddenly very abundant and nobody was quite sure why. This can be contrasted with the collapse of the North Atlantic cod stock in the 1990s. Alternatively, during this time, cod recruitment was very poor, and again, no one was quite sure why. So the current models for predicting recruitment mainly focus on spawning stock biomass. This means that they use the biomass of the fish that they currently have to determine how many there will be. So as you can see from these graphs, they're not entirely accurate, and the relationship is weak at best. I would also like to direct your attention to the top right graph. It's the North Sea haddock 1963 to 1996 Beaverton Holt model. In the top left corner of this graph, you can see how much greater the really great recruitment years are than the poor ones. So why do we want to predict recruitment? Well, like I've been saying, with the high variability of recruitment, prediction is very difficult. And if we were able to predict that a really great year class was coming, a struggling fishery might be able to remain open. Alternatively, if we knew that a very poor year class was coming, we could determine which species and stocks need to be fished less in the coming year. So in 1998, Ransom Myers published a paper titled "When Do Environment Recruitment Relationships Work?" This paper has sort of become the basis for our study. In this paper, he took 47 relationships between environmental variables and recruitment that were previously thought to have existed. He retested these data and found that most of these correlations did not hold. I would like to direct your attention to the red box that I have on the screen. The pluses mean that the correlation was still found to be significant, and the minuses mean that the correlation was not. As you can see, it is overwhelmingly negative. So this has become the main criticism of environmental variables being related with recruitment today, is that when you get more data, the relationship does not hold up. Despite this, it was found that in striped bass, poor river flow can in fact predict poor recruitment. This was found in 7 different stocks of striped bass. So the objective of my study was to determine if what Gross et al. Observed in striped bass was just a one-time phenomenon, or if this is a general pattern that can be applied to multiple stocks and species. I also aimed to determine a better way to predict recruitment, as the current models do not do a great job and are not super accurate. Finally, we wanted to determine if there is an association between extreme environmental variables and poor recruitment. So our null hypothesis in this study is that when a relevant environmental variable is extreme, recruitment is as good or better than when the variable is not extreme. Our alternate hypothesis is that recruitment is generally worse when a relevant environmental variable is extreme. So for our methods, we took the Myers 1998 paper disproving environment recruitment relationships, and we reevaluated these relationships, this time looking for indications that extreme conditions do in fact predict poor recruitment. We only looked at relationships that had previously failed. This is because if there truly is no relationship between these variables, then extreme conditions should not predict poor recruitment and our test should fail. So by doing this, we avoid any criticism of choosing stocks that we already knew would work. In our study, we defined extreme as the lower or upper 1/3 of values. We looked at the median, the standard deviation, and the proportion of good versus bad recruitment as metrics to evaluate recruitment. The reason we look at standard deviation so closely in the study is that it's a measure of the noise in a data set, meaning that it shows how predictable a data set is. So by looking at the standard deviation on the left and on the right, we can determine which side is more predictable. So in Myers' 1998 paper, he used several other sources as his retest data. So the sources that I looked at were the Brander 1992 paper, which examined if there was a relationship between Calidus or zooplankton abundance and cod recruitment in the North Sea. I also examined Chouinard and Frichet's 1994 paper, Hutchings and Myers' 1994 paper, and Surchuk et al.'s 1994 paper, all which examined temperature and cod recruitment in various stocks of cod. I looked at Frank et al. 1994, which determined if there was a relationship between river discharge and cod recruitment in Georges Bank. Finally, I looked at Myers et al. 1993, which determined if there was a relationship between salinity and cod recruitment in Labrador. It is important to note that when river discharge is high, salinity is low, meaning these last two are in fact related. You may have also noticed that all of these stocks pertain to cod. The reason for this is that with 47 different relationships, I did not have the time or resources to look at every single relationship. By choosing cod and selecting them each time, I was consistent and I was objective and selected stocks that, I was consistent and objective in my selection of stocks to use. So as I said earlier, correlations do tend to fall apart with environmental variables. The Brander 1992 paper is a perfect example of this. On the y-axis, we have recruitment, and on the x-axis, we have Calidus abundance. You can see the black triangles are the old data, and the red circles are the new data. So in the old data, it does appear to be correlated and appears to follow a trend. However, with the addition of the new data, the correlation completely falls apart and it looks almost random. So before I dive into my results and my graphs, I just thought I would orient you first. So the black line is our threshold for extreme, and this is the lower 1/3 of values. So that will be consistent in all of our graphs. And the gray dashed line is the median recruitment, above which we define as good recruitment and below which we define as poor recruitment. So for the Brander 1992 paper, we looked at cod recruitment versus calanus abundance. There is a reason to believe that these two variables would in fact be correlated. This is because larval cod feed on calanus. So as you can see on the left, the median is 271 and the median on the right is 426. This does show that recruitment is worse in extreme conditions. Additionally, the standard deviation is lower on the left than the right, which indicates that the left is more predictable. Finally, the proportion of bad recruitment on the left is 57%, and the proportion of bad recruitment on the right is 47%. All of this indicates that calanus abundance is in fact related to recruitment. Up next, we have the Myers et al. 1993 paper, which examined recruitment for salinity in Labrador. On the x-axis, we have salinity, and on the y-axis, we have recruitment. So the median is 29 million on the left and 47.8 million on the right. This follows the same pattern as the previous graph that we just saw. The standard deviation is 13.6 on the left and 20.8 on the right. This is markedly different, with the left being much more predictable than the right. The proportion of bad versus good recruitment is where we see the biggest difference. There's 78% poor recruitment on the left in the extreme conditions and 40% poor recruitment on the right in the normal conditions. This is a very strong relationship and shows that salinity does in fact cod recruitment in Labrador. Next, we have recruitment versus river discharge from Frank et al.'s 1994 paper. This is related to salinity because when we have a high river discharge, we see low salinity. So in this graph, extreme and normal are flipped because we would expect a low salinity to negatively affect cod, meaning we would expect a high river discharge to negatively affect cod recruitment. So on the right this time, in the extreme conditions, our median is about 25, where our median is 29.4 on the left. Oh, no, that's not right. Excuse me. The median is 20, oh, I'm sorry. And so as you can see, the median is higher on the left than on the right in cod recruitment. The standard deviation on the right is 5.6, and the standard deviation on the left is 6.3. This shows that the right is in fact more predictable than the left. There's 70% poor recruitment on the right and 41% poor recruitment on the left. This also shows that for salinity, which is consistent with the last stock, high salinity or low river discharge negatively affects cod recruitment. Up next, we have Hutchings-Myers' 1994 paper, which examines recruitment versus temperature anomaly. On the left, in our extreme conditions, the median is 228, where on the right, in the normal conditions, the median is 332. This follows the same pattern as the previous three stocks and shows that extreme conditions do in fact predict poor recruitment. The standard deviation on the left is 293.6, and the standard deviation on the right is 319.5. Additionally, we see 55% poor recruitment in the extreme conditions and 50% poor recruitment in the normal conditions, which is slightly less. Here we have an additional temperature relationship from Chouinard and Frichet, 1994. So on the left, in our extreme conditions, the median is 71.5, where on the right it is 101.5. This follows the same exact pattern as the previous graphs and shows that further reinforces that extreme conditions can predict poor recruitment. The standard deviation on the left is 33.2, while the standard deviation on the right is 52.5. The proportion of bad recruitment on the left is 60%, and the proportion of bad recruitment on the right is 42%. So at this point, we had 5 stocks out of 5 that reinforced our hypothesis that extreme conditions do in fact predict poor recruitment. However, we want to be objective, so we looked at every single stock of cod that we could. We came across Serchuk et al. 1994. And I would like to preface this by saying, when we read the paper, there was no clear hypothesis as to what they were saying. They looked at multiple variables, but Myers took it to say that high temperature was associated with poor recruitment, and he then disproved this hypothesis. So when I went to do this reanalysis, I could not conclude that the extreme conditions in this case were related to poor recruitment. So as you can see, the median on the right in the extreme conditions is 0.9, and the median on the left is 0.6. Both have standard deviations of about 1, which would suggest that they are equally predictable. Furthermore, the normal conditions have a higher percent of poor recruitment than the extreme conditions do. All of this might indicate to us that spring temperature, which is what they were looking at here, may not be a relevant variable for looking at cod recruitment. You could make the argument that if we flipped it, it might prove to be significant, but we wanted to be consistent and objective and stay true to exactly what Myers had done and not just look at data to reinforce what we already thought. Despite this, 5 out of our 6 data sets indicate that extreme conditions can in fact predict poor recruitment. Of these, salinity shows the clearest relationship, with both of those graphs looking like the extreme conditions are much, much lower than the normal conditions. This is enough of a trend for us to say that extreme environmental conditions do in fact predict poor recruitment. In 5 out of the 6 graphs, the median was lower in extreme conditions than normal conditions, as well as the standard deviation, meaning that in general, recruitment is worse and more predictable. Furthermore, the proportion of bad recruitment in the extreme conditions in 5 out of the 6 graphs was higher than in the normal conditions, which indicates the overall general pattern of the data. So for our implications, this does imply that certain environmental variables may have predictive ability for poor recruitment for cod. This could definitely be very helpful in predicting recruitment, as the current models are not super accurate, and this could be a way to better predict when those really bad year classes are coming. To continue to look at the poor recruitment hypothesis would be examining more species and stocks. Additionally, we could, now that we know that striped bass and cod follow this pattern, it would be important to know if other species and stocks do this, so we could apply it to more fisheries and more stocks. Another way that we could expand this would be to look beyond just the Myers paper and examine more current data to determine if this trend holds true today. I would like to thank everyone who made this presentation possible, especially my mentors, Dr. John Honig and Julie Gross, as well as the REU staff, Dr. Rochelle Seitz, Dr. Grace Massey, and Stephanie Peart. I would be happy to take any questions at this time.
All right, thank you so much, Annabelle. Are there any questions? All right, we have one in the audience from Nihal.
Okay, good job, Annabelle. That was good. I just had a quick, simple question. On your graphs, when you were talking about the extreme conditions like salinity and temperature, but specifically salinity, what was the measure for it? Like, what was it? Because it was like 1 point. I don't know. Can you go back to it?
Yes, I can.
Yeah, no, yeah, go to, yeah. So that one and river discharge, what was the measure for the x-axis? Like, is it just, like, do you know what I'm trying to ask?
Oh, no, I do know what you're trying to ask. And, you know, I'm not entirely sure, and I don't want to correct, but what's important to note when looking at these is you do see the overall pattern of extreme conditions being, having worse recruitment than normal conditions.
Okay, cool. And was the proportion, was that just, you took the recruitment below and above and just calculated that?
Mm-hmm.
Cool.
Yeah, so for example, you can see there's about 7 points below the median on this one, and there's 2 above, which would be 78% poor recruitment.
We have another question from Stephanie.
So with this slide up, because one of the things that stood out to me is your scale of your salinity changes. And we know that in the ocean, you can easily see within a day, just from a freshwater rain event, the salinity change more than just 1.5 PSU or parts per thousand. Are the cod and the recruitment that sensitive that just a 0.5 difference will impact poor recruitment versus good recruitment?
Well, I don't claim to be an expert on cod and salinity, but this is the data that we were provided with and the data that was originally used in the Myers 1998 paper, which is the data that we aimed to use. What I would expect based on my knowledge of extreme and normal conditions is that 0.5 difference might not make a difference if it's 32 to 35. We liken it to like temperature in humans. If it's 70 degrees out versus 80 degrees out, that doesn't make a difference. But if it's negative 30 degrees out that makes a difference. So I kind of liken it to that, where salinity is not important until it is. And so while it's difficult to pinpoint exactly where salinity becomes important, we just use the lower 1/3 to remain consistent and objective throughout our entire study, so we wouldn't be accused of curve fitting. But yeah, I do not know enough about the biology of cod to know if they are super sensitive to salinity or not, but that's a great question. Thank you.
For being early career, that's an excellent way of answering that question. Thank you.
Are there any more questions from the audience or perhaps in the chat? If you have a question, you can even just type question and then let us know what you want to ask. Excuse me. Oh, we have one in the front. Okay, so from Duncan.
Did great, but I just have a quick question. Why did you choose to do the median deviation from the median for these events instead of regression lines? Curve fitting you mentioned, Stephanie?
Okay, so we don't feel necessarily ready to model this yet. We're more just determining if if this pattern even exists, because we are looking at this under the assumption that it does not work. So we wouldn't really want to fit a model to that yet. That would sort of be a next step. And this is more determining if this exists. Okay. Does that answer your question?
It does.
Okay.
I have a quick question. So you mentioned spring temperatures in one of your graphs. So what would be like on an average spring temperature at that point? Do you have any idea about that? If that's out of your scope, no worries.
So I have no idea what the average spring temperature would be, but what we've been looking at is temperature anomaly. So that is how different the temperature is from the median temperature. So here it can be negative 1.5 degrees Celsius or positive 1.5 degrees Celsius above what it usually is. And that's what most of these papers use to look at it, to make it a little bit easier on the eyes to look at so it's not going everywhere. But yeah, so to expand a little bit on the spring temperature, most of these other temperature graphs look at just the median temperature for the year. And this one just looked at in the spring months what was the temperature. So that is why we think this graph might not be as accurate as the rest of them, and it might not have as much predictive power as the rest of them.
Awesome. Thank you, Annabelle. We are going to move on to our next presenter. Our next presenter is Amanda Peischer, and her mentors were Dr. Rochelle Seitz and also Katie Nick. She is also from Old Dominion University.
All right, thank you. So my project was on the assessment of oyster castles and grow tiles on eastern oyster recruitment and size. So as some people have mentioned before, oysters are ecosystem engineers, and they provide a habitat for a lot of various organisms. They're a food source, they filter water, and they also provide shoreline protection. But lately there's been a decline of oyster reefs due to anthropogenic causes mostly, such as dredging, overharvesting, such as negative harvesting methods, and disease. We've been looking at reef restoration as a goal to restore oyster reefs, and our goal is increased recruitment to restore ecosystem services such as water filtration, habitat, shoreline protection, ecosystem diversity, and we are also trying to restore their economic value, and such as less spending on water quality services and increasing the value of fisheries. So one of the ways that we are doing reef restoration is with alternative substrates. And the reason we are using alternative substrates is because normally oyster shells are a prime substrate that we would use for restoration, but because of their limited availability, two main alternative substrates are calcium carbonate substrates such as limestone and clamshell, and non-calcium carbonate substrates such as sandstone, concrete, and porcelain. So my objectives for this project was to assess eastern oyster recruitment and size on oyster castles and grow tiles. And my success metrics were based on oyster recruitment. So we're looking for high oyster recruitment, oyster size, we're looking for larger size, and benefits cost ratios, basically to see if you are getting your money's worth. And my hypothesis is that oyster castles will have greater success over grow tiles on oyster recruitment and size due to its vertical surfaces and lower cost per meter squared. So the first substrate we used were oyster castles, which are a non-calcium carbonate substrate made with concrete, crushed shell, and gravel. They have vertical surfaces, and the base area of it is about 0.30 meters by 0.30 meters, and they cost about $6 each, and they weigh about 30 pounds. The second substrate we used were grow tiles, which are a calcium carbonate substrate made with concrete similar to oyster shells. They have varied orientation of surfaces. The base area is the same as the oyster castles, but they cost about $50 each for each substrate, and they weigh about 15 pounds. Both of the substrates were deployed in May of 2019 in the Chesapeake Bay in 4 locations, and they were sampled October 2019, July 2020, and June 2021. And going from north to south on the map, there's Cherrystone, Mockhorn Bay, which is listed as oyster on the map, Lynn Haven, and the Elizabeth River. At each site, 10 oyster castles and 10 grow tiles were at each site randomly interspersed in the intertidal zone. And here's a picture of each of the sites. So Cherrystone and Lynn Haven were sandy, Mockhorn Bay was a mud flat, and Elizabeth was also muddy, but had lots of rocks. The way that we sampled each of the substrates was that we used a PVC quadrant, and we used a random numbers table to choose the quadrant that we would be measuring from. And we listed the shell height in millimeters of each of the oysters in the quadrant that was picked, the type of substrate, whether it was an oyster castle or grow tile, and the umbo needed to be in the quadrant for us to measure it, which is the hinge of the oyster. And we also stated whether the oyster was live or dead. At each site, we also took a YSI probe and recorded the temperature, dissolved oxygen, and salinity. And the individuals were summed to get the density per quadrant and converted to per meter squared. The 2 programs I used for this project were Minitab and Sigma Plot. Now, for each of my tests, I used 2-way analysis of variances, or ANOVAs, and I looked at the June 2021 data for the densities and heights, and the combined data, which looked at the 2019 data, 2020, and also this year's 2021. And I also looked at the densities and heights. The previous year's data were collected from previous individuals, but the data was given to me to analyze. Now, the water quality at each site, we looked at the temperature, dissolved oxygen, and the salinity. So Mockhorn Bay and Cherrystone, they both had high salinities, and between all of the sites, temperatures varied mostly due to seasonal, because we did not take samples at the same time each year. So my first analysis was June 2021, and we looked at live and dead density. Now, the reason we did live and dead is because live tells you what the current is, and dead tells you what has been. And there's also a possibility that the dead oysters serve as a substrate for incoming oysters. And so in the top right corner, I have listed the p-values of each of the individual tests. So for live density, site and type were significant, but the interaction for site and type was not. And for dead density, site was significant, and the interaction between site and type were significant, but type was not. In the bottom left picture, I've listed the live oyster density in meters squared by site, and almost all sites had greater live densities on oyster castles except for Mockhorn Bay. On the right graph is the dead oyster density by site and type, and we could not make a generalization of that because of the significant interaction. These graphs show the live and dead oyster heights for June 2021. So in the top right again, you'll see my p-values. And for live height, site and type were significant. And the site and type interaction we said was slightly or near significant. And I highlighted that in blue. For the dead height, site was significant, but the interaction was not. And most sites had greater oyster heights on grow tiles with the exception of Elizabeth and Cherrystone. In the bottom left, you can see the live oyster height by site and type. On the bottom right, you can see the dead oyster height by site and type. And I've listed some of the values there because they're really pretty close. So this data shows the combined, which again was between 2019, 2020, and 2021. So in the top right again, I've listed my p-values. So for live density, all the p-values plus the interaction were significant. And so I can't make a generalization about that. For the live density by year and type, the interaction was not significant, but the type p-value was. And so the live densities by year varied, but there was a significant year by site interaction. So this is also the combined data, but this one is the dead density by year and site and the dead density by year and type. So the dead density by year and site did not have a significant interaction, but the year and site p-values were significant. The dead density by year and type had a significant year p-value, but the year and type interaction was not significant. So the density patterns were almost similar by site among all years, and Mockhorn Bay had the highest dead density across all years. For the dead density by year and type, there was no difference by type, and there was a very minimal difference for 2020. These graphs show the live oyster heights by year and site and year and type. So year and site had significant p-values, and the interaction was also significant. The live height had significant p-values for year and type, but the interaction was only slightly significant. And there was a significant year by site interaction, and the live height was typically greater on grow tiles in all years. So these graphs show the dead oyster heights by year and site on the left and year and type on the right. And for all the p-values and the interactions, they were all significant, and so I can't make a generalization about them. And these are my histograms for all years and all sites. So on the right, I listed the frequency peaks for each of the years. And so on the top row, you can see the grow tiles, and the bottom row, you can see the oyster castles. Now for 2021, there were two peaks that it may be hard to notice, but I listed was 30 to 40 millimeters for both years and 60 to 70 millimeters for both years. And there was an average of 40 millimeter growth from 2019 to 2021. This here shows the benefit to cost ratio for the oyster castles and grow tiles. The benefit to cost ratios were calculated with the benefit per meter squared for each year, and the benefit was calculated from the total mean of live densities per year using the quadrant area of 0.15 meters by 0.15 meters and the cost per meter squared of the oyster castles and the grow tiles. And the raw cost of an oyster castle was $6, and the raw cost of a grow tile was $50. Now, but when I put it per meter squared, the oyster castles cost $64.58 per meter squared, and the grow tiles cost $538.19 per meter squared. And the ratio was calculated by the benefit over the cost. And as you can see, for every single year, the oyster castles benefited 10 times more than the grow tiles every single year. So oyster castles performed overall better than grow tiles. So for recruitment, oyster castles had greater live recruitment over grow tiles, and there were larger but for the heights, there were larger live oyster heights were on the grow tiles than the oyster castles. But as you could see from the histograms, there was a very good similarity between each of them. But the benefit to cost ratio is where you could see a major difference, as far as the oyster castles benefited 10 times greater than the grow tiles every single year. And I would like to acknowledge Dr. Rochelle Seitz, Katie Nick, the VIMS REU program with Dr. Seitz, Dr. Grace Massey, Stephanie Peart, our program funding by Grow Oyster Reefs LLC, and our program funding NSF. Any questions?
All right. So thank you, Amanda. We're going to take some questions now. If there are any questions from the audience or in the Zoom chat, feel free to let us know. Oh. We have a question from Nihal.
Okay. All right. My question was, so just as based off of what I was looking at, do you think you know what may have made the difference between, since all your sites, they were deployed out in 2019, but it looked like the Elizabeth River had way bigger oysters because of the measurements you said with the heights, but then like Bongkeun Bay and Cherrystone had way more density. So do you think there's a do you know what may have caused that difference in height versus density across the site?
Okay. I don't really know what the difference would be, but like I will say, we did notice that between all of them, when I ran the test, that it was just a major difference by site. So it could have been, because you could see the pictures, there was a variation as far as like landscape, but I'm not sure.
We have another question, and this time from Dr. Seitz.
So nice job, Amanda. So you showed that the cost-benefit ratio was better for the oyster castles, but you also mentioned that the grow tiles were lighter. They're 15 pounds
50%, yes.
versus 50 pounds. And so given that, do you think there may be an application for those grow tiles maybe in certain habitats?
I think maybe there might have been, because as you could tell, like the different weights that like some of them, when we went to the sites, they really got sunk in. So that could also have led to a higher mortality. As you could see, like Bongkeun Bay had a lot of oysters, but they also had very high mortality.
And that was the muddy site.
Yes, very.
I have a question. So it's going off of that. So if the castles and the tiles are sinking into the mud and they're causing potentially more mortality, is that also eliminating the surface area that the oysters can attach to?
I would say yes and no, because I think it's not like they're sinking right away. They're sinking over time. So I think there's a time period that they can attach to it, you know, but it's just a matter of how fast are they attaching. And it's like, are they attaching and then sinking, or, you know, is it sinking before they can attach? But I'm not sure. Yes.
Great job, Amanda. I have two questions similarly related, maybe. So the first question is going back similar to what Rochelle was asking about the growth tiles versus growth castles and the cost to benefit. The one being that the castles are cheaper, but they weigh more, and versus the tiles are lighter, but they cost more. What about in deployment because you have something that costs cheaper, but it weighs more? Have you taken into consideration the cost of fuel in deployment or manpower in deployment and calculating that into your cost per benefit ratio?
No, I did not. The benefit to cost ratio was simply just how much did it cost for the substrate and then how much benefit that we got from our sampling. But we did not take those costs into account.
Okay, good to know. Also, second question. So on your related to that, you said one of your slides, I believe, said that the castles had more live, but the tiles had larger larger oysters. So would there be a benefit that even though it costs more to make the tiles, but you're getting larger oysters recruiting on the tiles? Like, would that change your benefit analysis?
I think it more depends on what your goal is, as far as if you're looking for higher recruitment of live, or if you're specifically looking for larger. I guess it all depends on the project.
That's an excellent answer. And if I have time for one quick more.
Yeah.
So there is a big difference between your site locations on the amount of live oysters. Can you give us some idea or hypothesis of your own of why there's such a difference between like the survivability of the oysters on the Eastern Shore versus the Lafayette or the Lynnhaven and the Elizabeth?
I don't know if I could answer that. I mean, Lynnhaven and Sherryston, I think, were the most closely related, and Mockburn Bay and Elizabeth, I would say, were semi, but not completely. I don't know if I could answer that, though.
There is another question from the audience from Duncan.
Asked a question every time so far. I'm going to make sure I answer your question. Did a great job. I just had quick question. So you were out in the field sampling, right?
Yes.
Yes.
For 2021.
Yes. So are the oysters competing with, say, seagrasses for things you can? It's a little hard to see those photos, but it looks like there's some other things in the water.
Wait. So your question is?
Are there seagrass beds around where you were placing these castles?
Not that I know of. No.
Thank you, Amanda. We're going to move on to our next presenter. Our next presenter is Nihal Guennouni, and she is from Christopher Newport University. Her mentor was Dr. Ram Lipschitz and Gabrielle Saluda.
Thank you, Amanda. How do I make it show? All right, hi everybody. My name is Nihal Guennouni, as Amanda introduced me. Today I'm going to be talking about the project where I focus on the effect of reef height and oyster addition on persistence of experimental restoration reefs in the York River, Virginia. So I would go into a deep introduction of oysters, but as you've heard today, they are basically ecosystem engineers and they're crucial to the ecosystem and habitat around them. As you can see here, they do a lot more than just provide habitat. They also help reduce energy by protecting a lot of the shorelines, as the previous presentation emphasized. They provide a rich nutrient source for seafloor animals. They also help in denitrification processes, and they help clear water results with seagrass recovery. As you can see in the figure in the bottom left, this is a distribution of the eastern oyster across the east coast of the US, and you can see they are also located in part of the Gulf. Although it had been mentioned in previous presentations, there is a lot of ecosystem stressors that oysters are experiencing, which have led to a lot of their decline. Habitat degradation is a big one for oysters as they suffer from silt covering and sedimentation, which emphasizes a lack of settlement for a lot of oyster larvae. There's also a problem of a loss of spawning due to low density of oysters present in the water column, and a lot of times salinity can be too low or too high, affecting their survival over time. So there is an estimated 85% of oyster reef habitats lost globally, and as you can see in this figure here, by NOAA, the National Oceanic and Atmospheric Administration, of Chesapeake Bay oyster landings by state, so Virginia and Maryland, from 1880 to 2008. You can see that there is a pretty severe decline in oyster landings over time, indicating the importance of why restoration for these reef habitats are crucial. So jumping into a little bit of restoration, we saw that there are different substrates that can be emphasized for restoration efforts. However, here are some examples of just different methods of restoration. On the far right, we have spat on shell, where you are essentially just attaching younger oysters onto a previously dead shell and then transplanting that, onto another reef. We also have here in the middle diamond reefs, which are constructed of cement or concrete into a diamond shape and attached with either clam or oyster shells on the outside to attract oyster larvae and other inhabitants of a reef. The far left is a picture of a bunch of dead oyster shells which just get thrown into the intertidal zone of any coast, and they will keep accumulating until a reef essentially is formed. So you may ask, why is this important and why do we care so much about the specific methods of restoration? In order to assess the best effective methods for restoration, we have to be able to find some sort of measure for success. And so there are two main questions that scientists in this field usually ask. One, is the restored habitat going to persist over time? And two, are there sufficient densities of living oysters present in the restored habitat? The four universal metrics that are used to help assess these questions are reef area, reef height, oyster density, and the oyster size frequency distribution. For this project, we're mainly going to be looking at reef height. And then on the right, you can see a figure emphasizing reef height in meters. You can see anything above 0.25 meters is going to see a success in persistence over time. You're going to see settlement, growth, and accumulation of a bunch of oysters, which will then attract other species around. However, if you have any reef built below that 0.25 meter threshold, you are going to experience a lot of degradation. You're going to see sedimentation, burial, and a lot of reef loss, as well as a lot of oyster mortality. Reef height has been studied a lot in the past, and it's a very crucial driver for restoration. And so looking at the addition of live oysters as well as reef height is going to provide a lot of insight for future restoration projects, which is then jumping into our objectives for the study, which is to determine the threshold reef height for persistence and whether or not addition of live adults will reduce the threshold for reef height. So the hope is maybe we can build a reef that's a little bit lower if we are just adding some live oysters that will attract that oyster larvae. So for the explanatory variables for this project, we are going to look at, as I said previously, initial reef height, addition of live oysters, and the location of the reef. The location of the reef will be split up into two factors, block and shore and riverside. For the response variables, we are going to be looking at the final reef height, and we're going to be looking at rugosity, which is simply a measure of complexity of the reef. So for the experimental setup for this project, we essentially started off Rachel Wilson in 2019 around July. She was the one who decided on these 4 measures for reef heights and the 3 treatments of adults added. It was replicated. We had the 3 treatments of 0, which is no live oysters, 2 centimeters of live oysters, and 4 centimeters of live oysters. And we had 4 different heights at 7.5 centimeters, 15, 22.5, and 30. This made for 12 separate treatments that were then placed out to be replicated as a block to make 48 reefs in total for this entire project. So when I'm saying block, that's how far away they are from the breakwater outside of the VIMS York River area. And so block 1 indicates closest to the breakwater. Block 4 is going to be the farthest away. 12 treatments, 4 blocks, 48 total reefs. The shore and riverside just indicate the reefs that are along. If you can see, all the reefs that are going to be on this side of the blocks are going to be the shore side, and everything on the opposite end are going to be riverside. So for sampling methods, essentially we were just looking at our final reef height and rugosity for measurements. And so in order to capture the final reef heights, we used a leveled meter stick that was placed on top of the reef with one person underneath, making sure that the meter stick was above the reef that we were accounting for. This was ensuring that it would stay vertical when we are measuring, considering that the reef is underwater. On the right here, you can see this picture of me holding a chain link of 0.75 meters, so 7.5 centimeters, or 0.75 centimeters, sorry. And that was basically laid across the top of the reef to measure rugosity, which I will explain in the next slide for better clarity. So rugosity, since we are trying to measure complexity, it's just looking at the small scale variations of amplitude in the height of a surface. So basically, on the left here in this picture, everything circled in blue, if you were to lay a chain over that reef, you would end up with a very low rugosity, since rugosity is measured as the chain link length over the straight length of the reef. On the right here in the red box, you're going to see an area with high rugosity, because there is the oyster shell is very large, and you're going to see a lot of different tiny oysters down here. So then moving into my hypothesis, and rather than trying to disprove a single hypothesis for this project, we decided to set up a series of models that would then best fit our data after it was collected. And this is just a reminder of the variables that we're going to be looking at: final height, adults added, block, and the location. The adults added in the initial height was an interaction that we wanted to look at to see if they together would have an effect on the final height of the reef or the rugosity. For statistical analysis, we wanted to figure out our best fit model, and in order to do that, we did some linear modeling. We had AIC and weighted probabilities, nonlinear regression, and a regression residual analysis. So jumping into our results, you can see here, first we're going to be looking at the response variable of final height. We have final height on the y-axis and the initial built height on the x-axis. The color coding in the top legend here is the adults added, indicating 0, 2, or 4 centimeters. So we had expected at the beginning of this experiment to see, with higher initial reef height, we'll see a higher final height. And with more adults added, we'll see an increase in final height as well. However, we did not see that same trend when it came to addition of oysters on the reef. You can actually see it either decreased or remain consistent across the different initial heights. However, we did see an increase from the final heights. Then jumping into looking at the block effects on the reef height, since adults added didn't really have that effect we expected, you can see that the blue and the green, which were the two blocks farthest away from the breakwater, all those points are higher up on this plot, which just indicates that a lot of those reefs did see higher final heights and a growth from the initial heights. And a lot of the blocks that were close, the block 1 reefs, sorry, were closer to the breakwater and did experience some lower values in their final height. So since we have this data now and we see that there has been an increase from the initial to final height over the past 2 years, we decided that we would want to model our persistence, and we needed to figure out if it was going to be linear or nonlinear. And so just to clarify, your straight line is going to be your linear regression, and then the power curves and the sigmoid are examples of nonlinear regression. So for final height, we decided to run an AIC to compare our regression models. And it turned out that the sigmoid relationship was going to best represent our data. You can see here for the weights, it's at 0.83 compared to a low 0.08, 0.04, and 0.05 for the other models. Essentially, this just means that the probability of a sigmoid regression fitting the data is going to be your best option. And as you can see here, you have the final height on the y-axis and the initial height on the x-axis. Around 6 to 7 centimeters is going to be your threshold for your reef height. Essentially, what does that mean? Around 6 to 7 centimeters, you're going to start to see a positive increase in the height. Now moving on to rugosity, we did not see a clear relationship in our data. However, when we ran the AIC, it turned out that a linear regression would be the best model to represent our data collected. It is pretty scattered, but we're going to then run an AIC again with linear models to figure out which variables from the beginning are going to affect the final rugosity. So as you can see here in the right column in the highlighted boxes, 0.7 for our linear model 3 is going to be the best representation for rugosity. What does this mean? Just to remind you all, linear model 3 was rugosity as a function of the final height and the adults added. So this is pretty cool, because in our final height, we did not see any correlation with our adults added, but for rugosity, we can see that this produced the strongest model. And so to explain it visually, our pink is going to be the 0 centimeters, so none. Orange is going to be 2 centimeters, and blue is going to be 4 centimeters. As you had more adults added, you did see higher values for rugosity. And you can see the pink, a lot of them were very close to 1. And essentially, when you see 1, that means that the straight length and the bumpy length were essentially the same. So based off of all of that, we can see that initial reef height is a major driver for persistence, where we see 6 to 7 centimeters is a minimum threshold for persistent reefs, and that the addition of live adults, although had no impact for initial reef height or for final reef height, it did for rugosity. This is important because we have seen a lot of studies in the past talk about the importance of making sure you're building your reefs at a adequate height, making sure you're not building them too low, otherwise sedimentation will take over and you'll see a lot of mortality. But being able to see that the addition of live adults did impact the complexity of the reef is very crucial for future projects, because more complexity means that there may be an increased survival amongst the species, including the oysters on the reef. So just since we touched on future implications, it would be better to look at rugosity on a larger scale. So rather than just looking at our 48 reefs and just over these past 2 years, maybe making this a longitudinal study that we could explore over time and over more reefs and maybe more sites as well. We also could look at oyster density as a measure of persistence, since that is one of the 2 major questions you must ask in restoration. Although with the scope of this project, we didn't have as much time and resources to look at oyster density. So we settled for our reef height because we know that that's a major driver. Also, looking at live and dead measurements to observe proportional survival and mortality rates would be beneficial, just to see, are these reefs actually persisting, or is everything on the reef dead? Community structure as a function of reef height would be beneficial as well, being able to see if there is an increase in biodiversity across the reef with a lower or higher reef height. And so I just want to say thank you to, I just want to say thank you to my mentors, Dr. Lipschitz and Gabby Saluda, the NSF REU program, including our program director, coordinator, and our TA, and the rest of the Lipschitz and Seitz lab for all of their help with this project and, just the support they've given throughout this whole summer. So if anybody has any questions, I'll take them now.
Great job, Nihal. All right. So if there are any questions from the audience, or as I mentioned before, you can throw them into the Zoom chat and we would be happy to answer them. Oh, we have a question from Dr. Grace Macy.
Great job. Can you go back to where you were showing your increased heights from block 1 to block 4?
This one?
Right there, yes. Yeah, so why do you think there would be more increase in blocks 3 and 4 compared to 1 and 2?
Okay, so that's a good question. I personally think that blocks 3 and 4, since they were farther away from the breakwater, they're farther away from a lot of activity, whether that's people entering the shore, coming off. Just in general, it being farther away from the breakwater allows maybe better flow. And so with better flow might be better opportunities for the oysters on those reefs to obtain the food they need, since they are filter feeders, and with less distractions, maybe there is, or maybe with less I guess distractions are just interference from outside sources. There might be lower turbidity in that area so that they are just thriving better. Again, it's not a super strong relationship here where it's significantly 3 and 4 way higher. So that could definitely be explored if we had maybe had more reefs.
We have another question from Dr. Seitz.
Nice job, Nihal.
Thanks.
So could you go back to your figure on the final heights with adults added, this one. So I guess it was surprising that the adults added didn't increase your final height, and in some cases may have decreased. So can you give an explanation as to why that might be happening?
Okay, so I personally don't know why adults didn't add. I genuinely thought that maybe more live oysters would help the reef out, that even if the reef is suffering from sinking or anything, that those live oysters on the top are gonna hold it together and allow for more settlement. Maybe the problem is, since they are just the live oysters at the top, maybe they're more prone to maybe predation, or maybe they're open to maybe they're just more vulnerable being there, settling all at the top, so that if they do suffer from some sort of removal or they do die, that below them there isn't much left. It's just dead shells. So maybe that just started to sink too. I don't know. That's the best answer I could give you.
Okay, thanks.
Oh, we have a question from Stephanie.
Great job, Nihal. So I have a question. I have two questions, and one is related to what Rochelle just asked. So with respect to the live oysters on top and it not increasing your overall final height, one, did you guys do any sorts of weights or measurements? Do live oysters weigh more than dead oysters? That would cause some sinking that would automatically decrease the height. And two, did you do any sort of like what Amanda P did with counting live versus dead oysters at, like, so were all of the alive oysters that you put out, did they stay alive at the end of your experiment?
That's a really good question, and that's a question that we have ourselves doing this study. So that's why I was saying that it would be important, like the next step in this project would be to be able to pull up those reefs and take live and dead measurements, assess, you know, are they bigger, are they smaller, are they all dead, or are they alive? And so we really don't know, since they had been out there since 2019 and then they had just been looked at again this year. They might have just died maybe after like a month being out there. I don't really know, but that's a question that we were asking ourselves and something that we need to look further into. Did I answer all your questions?
The first one was, how much does a live adult oyster weigh more than a dead adult oyster?
I'm not oyster expert, but I believe they'd be heavier just because they actually have contents on the inside and digestive contents. So I would assume they'd be heavier. But then again, I don't know, maybe they aren't, but I think they are.
All right, thank you, Nihal. We are done with all of our presenters for today. So I'd like to welcome Dr. Seitz back up to the stage for some closing remarks.
Hey y'all, this is Ram Lipsius. I just want to make a quick comment, and that is that sorry I wasn't able to attend in person. I had a webinar to attend, but I wanted to thank Rachel Wilson, actually the one who designed the experiment and set it out. She's actually in on this Zoom meeting, so thanks for being here, Rachel, and interesting results from your initial experiment.
All right. Well, with that brings to a close our presentation of research results for today. And I just wanted to say, wow, that was amazing what you guys have come up with in a short span of 10 weeks of research here at VIMS. So another round of applause for all of you. Excellent job. And of course, I need to say once again, thank you to all the mentors for guiding these students through the research for over these 10 weeks. I know it's been difficult dealing with Zoom and a little bit in person when possible and rolling with the punches and all the changes. So thanks to all the mentors for sticking with it and doing a great job getting everyone through. And also wanted to say thanks to the support system at home, because normally you would just be here with us and you wouldn't have to rely on parents to give you time away and quiet time to work on your studies. So have to say thanks to the support system at home as well. And then I definitely wanted to also thank Grace Massey and Stephanie Peart for helping out and making this all possible. So big round of applause for you guys. And I also wanted to invite all the mentors and anyone who's listening who might be on campus to join us for a short reception in the Waterman's Hall lobby. And just wanted to say congrats to all the students. Nice job. That's it. Thanks for coming, everyone.